Så det var inte riktigt när fonden startade, vilket var, eller grundades 1989. So now I also like to say welcome to all of you who are wherever you are in the world. And if you don't understand Swedish, all the presentations will be in English. But the kind of talk now I will do is be in Swedish, and the panel debate later also in Swedish. Så mitt namn är Christer Fuglesang och jag är ordförande för Göran Gustafssons stiftelse för medicin, naturvetenskaplig och medicinsk forskning. Långt namn på en fin stiftelse. Och jag måste säga att när jag kom in i kontakt med den här stiftelsen så blev jag väldigt imponerad av framförallt då Göran Gustafsson, som alltså växte upp i en liten by söder om Gällivare som någon mittenbarn av familj med 14 barn. Fick aldrig någon möjlighet själv att utbildas ordentligt. Bara 6 års skola. Men var definitivt en entreprenör och affärsman och kunde steg för steg skapa sig själv en ordentlig förmögenhet. Men han kände alltid liksom att han hade fått så mycket från samhället. Han ville ge tillbaka det. Och det var så han uttryckte det själv. Liksom, att samhället hjälpte mig till framgång, jag vill ge tillbaka. Och han brann för forskning och han förstod hur betydelsen för forskning för att liksom bygga ett bättre samhälle och bättre framtid för mänskligheten. Så därför då så inte bara en stiftelse utan 2 stiftelser grundade han. 1986 forskning för teknik och medicin vid KTH, Uppsala universitet, och 3 år senare den här stiftelsen då. Som då 1989 han donerade 270 miljoner kronor till ungefär. Som under åren har växt. Och det beroende på vad aktiekursen är, dagens läge är, så är det i alla fall värt betydligt över 2 miljarder kronor idag. Och det är också tack vare att vi har en väldigt bra förvaltningsenhet för stiftelsen, G-förvaltning. Under nu då 31 års tid, i år var det första gången, så har det delats ut totalt över 600 miljoner kronor. Nu är det 27 miljoner kronor per år som delas ut till 5 unga forskare. Unga i sammanhanget betyder man får inte vara äldre än 45 år. Ja, nu börjar jag bli så gammal så jag tycker att det är ung då. Det kanske inte tyckte på den tiden. Men, och det är 5 områden. Det är matematik, det är fysik, det är kemi, molekylär biologi och i medicin. Och det är en del som är personligt pris. Det är runt 250 000. Men den största delen då, alltså det är över 5 miljoner kronor som forskarna får använda till fri forskning. Och det här skulle jag liksom vilja trycka på. De får göra vad de vill. De behöver inte bekymra sig om att det ska skrivas någonting hit och dit och ansökan. Utan det är verkligen en guldgåva för den som är forskare och vill försöka prova på saker då. Och över åren så är det många framstående forskare som har sedan gjort ännu mera bättre karriärer. Och vi har till och med en som fick Nobelpriset nyligen då, Emmanuel Charpentier. Tyvärr hon som väldigt efterfrågad, så hon hade inte möjlighet att komma hit idag då. Den här stiftelsen då, den har en styrelse, 9 ledamöter. Och 5 av dem utses av KVA, Kungliga vetenskapsakademin. Och de representerar var och en av de här 5 områdena. Och det är också en väldigt styrka i det här stiftelsen och priserna. Att det är KVA som liksom då, gräddan av forskningen vi har här i Sverige, som liksom bedömer vilka är de mest bästa pristagarna som de då nominerar till styrelsemötet. Där sen ingår 4 till som utses av Göran Gustafssons ättlingar. Så att det är fortfarande väldigt mycket Göran Gustafssons ättlingar som är med och påverkar den här stiftelsen. Och det var genom en av dem, Per Gustafsson, en av Göran Gustafssons söner, som kontaktade mig för några år sedan och undrade om jag inte kunde tänka mig att bli ordförande för den här stiftelsen. Och det var en väldigt trevligt och hedersamt uppdrag som jag är väldigt glad att ha. Per och jag känner varandra från KTH där vi pluggade ungefär samtidigt under flera år och sen goda vänner ända sedan dess då. Det här jubileet, det togs upp första gången så att jag kan komma ihåg det då, i alla fall på styrelsemöten vi hade 2019 utav Sara Gustafsson, en barnbarn till Göran som också är här. Där som också sitter i styrelsen. Och hon hade kommit på att det är 30 år sedan den här stiftelsen skapades. Instiftades. Alltså. Ja, det kanske är svårt att hinna med jubileum nu då. Men vi kan ha 30 år utav första utdelningen år 2021. Det har vi tid att planera för. Så det gjorde vi. Och planerade för det till september 2021. Ja, alla vet ju vad som hände. Och sen har vi haft ytterligare 3 datum till som vi har försökt få till den här jubileet. Så här är 4:e datumet faktiskt. Så äntligen så har vi det här nu då. Vi är så glada att det äntligen har kunnat bli av dem. Så det är ett års försenat 30-årsjubileum utav det första prisutdelningen. Så dagens program då. Vi kommer nu här i Beijersalen under eftermiddagen ha 8 pristagare som berättar om sin forskning. De kommer prata ungefär 15 minuter. Och sen har vi 5 minuter till frågor. Och jag hoppas att du kan få intressanta frågor och lite diskussion runt deras föredrag då. Det kommer bli en paus 23:03. Då kommer det serveras fika och lite snacks där ute. Och om ni inte redan märkte det när ni försökte komma in här så pass på att påpeka att den här fina salen, ni kan inte ta in fika eller grejer in här. Det kommer efter de 8 föredragen, vi kommer ju ha en paneldebatt där vi ska diskutera liksom betydelsen av sådana här priser som Göran Gustafssons stiftelsen delar ut och forskningens roll i framförallt kanske då hur privat forskningsstöd förhåller sig till det statliga. De frågorna ska vi ta upp. Klockan 5 så avslutar vi här idag. 2 timmars paus, rast. Tid att klä upp sig ännu lite bättre. Inför banketten klockan 7 i Stadshuset. Ni behöver inte vara 7 prick. Men från 7 så kan ni få en liten välkomstdrink och lyssna på lite trevlig musik. Men ni bör definitivt vara där 7:45 för då intar vi middagsborden i Gyllene salen. Då. Och där blir det ytterligare lite tal med en del musik. En av talen kom förut Per Gustafsson berättade om sin far. Och förstås jättegod mat och dryck. Har ni frågor sen så finns det de här värdinnorna i foajén och sådär som ni kan fråga om var vara månde så att säga. Och med det så skulle jag vilja faktiskt bjuda upp den första föreläsaren. So now I switch to English. Are you ready? Was I too fast here?
No, I'm ready.
Så vi har Pernilla Wittung Stavsede. She's a professor in chemistry at Chalmers, and she got the prize of the foundation in 2009 in chemistry. Please.
Thank you very, very much for that introduction, Christer, and for setting all this up. Oh. I'm so happy. I'm sure we're all so happy to have this actually happening, right? We waited 2 years and I updated my presentation so many times for this. But now you're going to hear it. So, I have to click. Yeah. See. So I will talk about my research around proteins, give you an overview of my scientific career and research directions in 15, a little bit more minutes. And here's a picture from when I got the prize in 2009, as you heard in chemistry, and my fellow winners that year. So I will take you from living organisms, human beings, animals that you can see. They all consist of many, many cells, millions and millions of cells. In those cells and outside are proteins. And proteins are the workhorses or molecular machines of life. And this is where my research centers and what we will talk about. But first I want to tell you my background, how I ended up in the position I have today at Chalmers. So I'll show you a map. So I was born in Umeå, long time ago. I moved to Gothenburg when I was little, and I got my education at Chalmers. I got a PhD in physical chemistry. Then I moved to America to do a postdoc at Caltech outside LA. And I was there for 2 years. Then I got hired at Tulane University in New Orleans as a professor in chemistry. And I spent 5 years in New Orleans. Then I was recruited to Rice University in Houston, Texas. So I spent another 5 years there. Then we decided to move back to Sweden. And Umeå University hired me as a professor in chemistry. I spent 7 years in Umeå, but then since 2015, I'm now back at Chalmers. And really, I moved back to Chalmers because they started a new department in biology and biological engineering. And my recruitment was part of building up that new department. We're going to talk about proteins. And proteins, most of the time, must fold in order to function. So, as many of you know, DNA, the information that we have in our cells, are transcribed to RNA that's translated to a polypeptide chain. So proteins are long chains of amino acids, a polypeptide chain, but these chains need to fold into a unique compact structure in order to function. That makes the active protein that can do its job, whatever that is. If this polypeptide chain misfolds, that can lead to aggregation or degradation, and you don't have that function. And there can be problems with protein folding because of mutations in DNA that will place the wrong amino acid in a particular position in a protein that doesn't fit with a compact structure. Or you can have external perturbations, say stress and aging, that affect folded proteins. This process of protein folding occurs spontaneously in our bodies now. You probably eaten lunch recently and proteins are made in your bodies from the amino acids that you got from your food. So you go from this unfolded state, kind of a boiled spaghetti-like structure, that's released from the ribosome. Then that immediately folds up to the active yarn ball-like structure by itself. Because all information for how to fold is encoded in this linear chain of amino acids. And the question is really, and has been and still is, how does a floppy boiled spaghetti know what structure to fold into? How does this work? And we know that folding is fast. It occurs on biological timescales in us all the time, from micro to milli to seconds. If it were a random process, it would take forever because there's so many degrees of freedom. So there must be pathways or guiding factors. And we want to figure those out. And by studying different protein systems, model proteins, as you can say, looking at speed, mechanism, energetics, intermediate structures, during these reactions, we can learn about them. Most of the time, folding works. But sometimes we have problems with folding, and that can cause human disease. So problems with folding can lead to loss of function, as I said. If the folded protein is not made properly, it cannot do its function. This is often the case in cancer. Tumor suppressors are not made properly. They cannot suppress tumors from spreading or growing. But we also have gain of function type of diseases, amyloid diseases, where proteins come together and aggregate into ordered fibers with a cross-beta sheet structure. This is the basis of many diseases that we know of today. Alzheimer's, Huntington's, Parkinson's, type 2 diabetes, prion diseases, ALS. In all these cases, a particular protein, a different protein in each case, comes together with its fellow proteins and form these long amyloids, which in all these diseases have the same overall fold, this amyloid fold. But we know of about 50 or so different amyloid diseases today. And we can see them under the microscope at these long strings if we want to study them. But these diseases are a huge problem. Age is the largest risk factor, and the population is getting older. So we're just going to see more and more of these diseases. And we have no cures. We can only treat symptoms for a while. That's as much as we get today. And to understand why it goes wrong in these cases, a first step is to understand correct folding. So how does correct folding work? Well, during the last 20, 30 years, many protein folding studies in test tubes in dilute solutions have been performed. And we have learned, I contributed to this early on in my career, we have learned a lot about mechanisms and speed and so on for how individual proteins fold. And for example, I have the world record in fastest folding protein, if that's something to have. This moved a little bit, okay. Most proteins are more complex. We need to also consider more complicated proteins. So we need to consider protein-protein interactions in case of oligomeric proteins that have more than one polypeptide chain. And many proteins do, especially in more advanced organisms such as humans. We also need to consider binding of metals from metal binding proteins. I don't know why this picture is, on top of my text here, but that's okay. I'll say what you need to hear. Metal binding proteins are important. Almost half of the proteins we have in our bodies bind a metal ion in order to be functional. Copper, zinc, iron, for example, are essential metals that we need to make proteins more active and do certain duties. But free metal ions are toxic most of the time. So cells have to be very careful how we deal with the metal ions that we need and place them in the right proteins. So these two more complicated features of proteins became my research area when I started my own research group. So I want to give you one example so you understand what I mean. If you think about blood, it's red. It contains hemoglobin, which is a protein that has 4 iron groups, so metals, and 4 polypeptide chains in its functional state. So it's an example of both these two complications, as I say. So our studies of many protein systems over many years with different cofactors, different metals, different shapes, different assembly status. From those, we have learned that protein-protein binding can precede folding for oligomeric proteins. So unfolded proteins can meet and that can trigger folding. We have found that metals can bind before folding and that affects how the folding reaction goes. So if we want to understand correct folding, we need to take these factors into account. Also, we often want to understand how proteins fold in real life in a cell. Then we have to remember that cells are highly crowded with many biomolecules. It's a mess in there with lots of proteins and other biomolecules packed together. So it's very dense. So it's not this dilute test tube solution that we like to use in our experiments to get quantitative data out. So early on, we pioneered how to study crowding in a test tube to mimic this cell environment. And we've learned that many properties of proteins can be affected by this crowded environment and the steric effects due to that. So these results led us to pose new questions. How are metals delivered to the right proteins without toxic side effects when you can bump into so many things? And what unwanted interactions can happen in this type of environment between proteins that maybe can promote disease? So over the years, my research has gone from focusing on protein folding principles, biophysics, trying to learn mechanisms, to coming more to protein function and dysfunction to understand that. So in the lab today, and maybe for the last 10 years since I got the Göran Gustafsson Prize, in a sense, there's 2 directions in the lab. One is about how copper ions, one of the essential metals, are moved around in the cell to reach the target protein that needs it for function. How do a cell move around the copper safely without any toxic effects? And this has led us into cancer. And the other direction is to go from folding to misfolding. And here we focus on amyloid formation and how that can be modulated by cross-reactivity with other biomolecules that you would interact with in a cell, and how that affects neurodegenerative diseases. So I'll just give you a glimpse of discoveries that we have made so you can get a feeling for what type of questions we are addressing, coming to breast cancer and Parkinson's disease. So as I said, copper is essential, but we don't have any free copper in our cells. Instead, we have dedicated proteins that move the copper around. And one key player is ATOX, a copper chaperone in the cytoplasm. So we try to study this protein, how it moves the copper, gets the copper, moves it to the next protein, and then eventually to a protein that needs it. We looked at elementary steps in the actual copper transfer event between two proteins. We looked at how the cancer drug cisplatin, which is also a metal, can hijack copper transport proteins and escape out of a cell, which is something you don't want if you take this as a drug. We looked at domain-domain communication because some of these proteins are large, they have many domains, and how do they interact with each other? And we looked with other partner proteins, discovered new partners that weren't known before that might also get copper this way. And we looked at mutations, for example, that lead to copper overload disease. And this led us into cancer because we know that copper levels are high in cancer cells, and that's, if there's no free copper, that means that copper proteins must be important. Which proteins are important in cancer? Which copper proteins? So we tried to look at that first from a systemic biology type of approach, looking at up and down regulation in different cancers of all the known copper proteins we have in a cell. And then we zoomed in on ATOX, the copper chaperone, because it's upregulated in breast cancer and in several other cancers too. But here we went in to figure out how this works. And we could find that ATOX promotes cell migration, how the cancer cells move. And this is important because this is in a way the first step of metastasis. A cell has to move to another place. We also took it to the patient level because we looked at patient survival, and we could see that patients that have high amounts of ATOX protein, they have less survival than patients that have low ATOX. So we think that maybe copper proteins can be targets for blocking cancer metastasis, or at least biomarkers for when to use copper chelation therapy. When we come to misfolding, we focus on alpha-synuclein, which is a key protein in Parkinson's disease. And here we started out, we stumbled on this really because we looked, I moved to Umeå and we collaborated with organic chemists to look at small molecules, how they can tune protein folding, correct protein folding. But then that didn't work, so we tried it on an intrinsically disordered protein, alpha-synuclein. And then we started to get really good, interesting results. So we tried to tune the reactions with these molecules as a way to identify and learn about intermediate species that we could accumulate. We also looked at interactions with membranes because synuclein might function in vesicle transport and how lipid chemistry can affect these interactions and how they cause aggregation. We looked at interactions with DNA because synuclein apparently is in the nucleus as well and can maybe play a role in gene regulation. And we tried to find a molecular link to type 2 diabetes because these patients are prone to get Parkinson's, but it's not clear why. And we found two proteins that are key players in type 2 diabetes that interact with synuclein in a way that could trigger the disease. And we also tried to look for ideas how Parkinson's start or how amyloid formations start. There are ideas now that the gut microbiome, external factors, affect or start these diseases. So we looked at proteins from bacteria in the gut. There's several proteins in our guts that also form amyloids, and that's part of biofilms many times. How do they interact with synuclein? And we've seen proteins that can both accelerate or block amyloid formation. Then we stumbled on a fish protein. You know, fish is good for the brain, right? But why is that? We found an abundant fish protein that gets into our bodies when we eat fish because it's an allergen. So we know it survives the gut. It blocks synuclein amyloid formation. It might be a new way to learn from in order to make future cures. It's found in most fish. It's always good to eat fish. But many things, external factors, can affect amyloid formation. We also connected back to copper in my last few seconds here. Because synuclein, this amyloid protein, is a copper binding protein. And we realized that it can get copper from this copper chaperone that I talked about around cancer, and they can form a complex. And to the point is that when you have Parkinson's, copper levels are lower in your cell. So maybe at those conditions, you abolish this type of complex, and the synuclein protein is free to aggregate. So clearly, synuclein has many interaction partners that modulate both function and amyloid formation. And we need to look at them one by careful experiments to understand, but then we have to put this whole puzzle together. So I'm not going to say much about what we do today, but I'll give you just a teaser. Just 1 minute. Yeah. And it's red here. Yeah. I just want to tell you kind of what we're looking at now to give you an idea. We want to understand, or try to understand, how amyloid fibers get longer. Because they extend and become longer and longer, then they can spread to other cells. But if we understand the mechanism, how new monomers are added onto these growing fibers, we would know more about how to make inhibitors. Also, we stumbled on a new property of amyloids. They're toxic, yes, but they're considered dead-end species. But it seems like actually amyloids, these fibers, can have catalytic activity and be enzymes doing chemistry. And that will be a new direction that people haven't thought about before. And we started to look at another very important cancer protein that people have claimed is important in cancer. And we found that, in fact, it binds copper. And it seems to protect against redox activity. So maybe this has something to do with this role in cancer. And this might be a new way to target this protein. So overall, if I think about what I'm doing, I try to reveal new molecular knowledge about proteins and their actions that may be used to combat human disease. It's fundamental studies, but still the goal is to, in a way, help humanity. Finally, very important, I want to say I don't do this alone, right? I mean, all my students, colleagues, friends, different places have been, different environments. That is very important. So nothing would happen without my students. So this is all the students I've had in my lab so far. Some got their PhD, others are postdocs or researchers. This is my current research group. We're kind of small right now, 5 people, but that's because I spend a lot of time on a gender initiative at Chalmers. Also something that's important to do. So my group has to be a little bit small for a while. And funding agencies, important. Previous funding agencies, Göran Gustafsson, that got a lot of these things going. And current funding, Wallenberg, that supports most of what I do, and the others help. And with that, thank you very much.
Thank you, Pania. Very exciting.
Thank you.
So, there's time for questions, are there?
Well, thank you for a fascinating talk. And I'm a physicist. I don't know anything about this, but I was intrigued by copper. I mean, you seem to play a very important role here. Can you elaborate why you think copper is sort of a key player?
So in cancer?
Well, you mentioned it several times.
Yeah. No. So I mean, we all need copper. So it's important in very fundamental reactions in our bodies. I mean, how we generate energy in respiration, in mitochondria. There's a lot of enzymes that need copper to do basic function, to kill redox species or to help cells move around the molecules. So, I mean, you have this essential role. So if you were to have no copper in your body, you would die. But then when it comes to cancer, because cancer cells grow so much and they divide and they spread and they're very active, they need more copper because a lot of these fundamental processes that is good for us are needed even more in a cancer setting. So that's why I think this increased demand of copper can be something, if we can tune that, we can kind of block cancer cells from growing. But the most important function of copper is in respiration, in cytochrome C oxidase, in the, those chains generating ATP and energy in the body.
A general question about misfolding. I guess essentially all sexually reproducing organisms are genetically variable. And when it comes to genetic variation in protein coding genes, some mutations cause non-synonymous changes, that there's an amino acid change. And most of those changes are deleterious in one way or the other. Mm-hmm. Do you think that the means means for deleteriousness is via misfolding, that there are problems with the kind of three-dimensional structure because of a non-synonymous substitution.
I could think, yeah, because otherwise that's the way to recognize that something is wrong, right? With that mutation, in a way, that protein is not doing what it can, and therefore the organism doesn't survive. It could be stability. It doesn't have to be pure misfolding, but it could be, yeah, you're not stable enough, so then under some conditions you don't do what you should. Of course, sometimes it could be a mutation that affects just an active site, and then you directly have no function. But I think a lot of it has to do with stability and therefore misfolding.
The question.
Yeah. You mentioned the misfolding, the plaquettes, in connection with Alzheimer's. Did you also mention it in connection with type 2 diabetes?
Yeah, you have
and so that, you know,
yeah.
So the question is, can this tell us something about possible relation between these two diseases? Because to my mind, as a physicist, they're completely different and unrelated, but perhaps not.
No, so Alzheimer's, type 2 diabetes, Parkinson's, I mean, they have this is really the same, that they have a protein that starts to form amyloids. Then the position of those plaques can be in the brain or in the pancreas, depending on which disease it is. And then the question is that we don't really know is how you start this. What is the trigger to get to these proteins forming these amyloids?
I meant, could it be that if you have type 2 diabetes, that means that you have a higher risk for getting Alzheimer's, because the body can make these plaquettes, and
yeah,
they might
not exactly, but what can happen is that, and this is known at least from test tube experiments in many cases, that amyloids on one protein can trigger amyloid formation of another protein. And that's what we see, for example, with type 2 diabetes, that IIPP protein that forms amyloids in diabetes. Those amyloids can start or get synuclein to form amyloids that would mimic then Parkinson's. So this cross-reactivity, and I mean, most proteins are expressed in almost all cells. So Parkinson's, that affect brain cells, but this protein, synuclein, is expressed a lot in the whole body. Actually, we have a lot of it in red blood cells, and nobody knows why. But it's also there in the gut, it's also there in the pancreas. So different proteins can meet and they circulate, so they can move around. So there's definitely connections, and there's generics in this, in terms of this amyloid fold, that many proteins, in principle, all proteins in some condition, can adopt that type of fold.
Okay, sista fråga. Last question.
Yeah, so you described the effect with the mammary tumors where you have copper binding proteins that protect against loss.
Mm-hmm.
So is this a primary effect? I mean, do the tumor accumulate mutations that affect the protein folding and thereby survive better and protect against loss better? So is it mutations in the tumor cells affecting the protein folding to enhance the copper binding and then protect? Is that the mechanism of this?
No, I think it's, I mean, so okay, of course you have a lot of mutations when you get a cancer, but the idea is, and we look at cancer cell lines in our lab, where we try to up and down regulate protein and see if they play a role in these different processes. But it seems in cancer cells, in general, you have higher redox reactivity because you just have more activity going on, and therefore you can have more redox also, and that's helpful to some degree. But if you go too high, I mean, you're on the balance there. So then it might be that this protein that I said could protect against redox by binding copper. Maybe that's a way to have protection in a cancer cell that's on a really high level of activity here and potential danger. And then you can chelate the copper and block redox to kind of survive in a way. But then if there's mutations in proteins that come to this, I don't know. Because that's not the way we looked at it. Maybe. Let me think about that.
Well, this is a really exciting and interesting and important subject. But you have to kind of go to the next speaker, but let's thank And then I would like to ask Peter Brodin to take the stage. He's a children's medical doctor and professor in children immunology. I hope I got that correct in English.
Yeah.
At the Karolinska Institutet, and he got our prize in 2019 in medicine.
Yeah. Actually 20. It was an error,
sorry.
but it's
Thank you for correcting.
recently. Thank you. Thanks so much. It's great to be here, and I'm really happy to share some of the work. So what I'll try to do is, first of all, I'm very grateful to the Göran Gustafsson Foundation, especially this freedom of using these funds for any kind of project. And the reason for that is because, as all of you know, who are active researchers, whenever you do something, you stumble upon a finding that you want to pursue. And to be able to do that and have some funds to address it without waiting 2 years for a VR grant and review process and all that is really, really incredibly valuable. So I'm very, very grateful for that. So what I'm going to talk about today is the work that we do in an area that we and others call systems immunology, or in my case, human systems immunology. So what I'll try to do is to introduce what we mean by this, because it's just one of those fancy words that people throw around without really explaining what they mean. And as you already heard, I'm a pediatrician working at the Karolinska Institute and University Hospital, and then I spend a part of my time at Imperial in London. My career is not as long as illustrious as Pernilla's here, so I didn't pick a slide for it, but I can tell you briefly in 2 sentences that I studied medicine and did all my clinical training at Karolinska Institute, and then I did my PhD there as well within an MD PhD program. Then I did a 2-year postdoc at Stanford University before I was recruited back to Karolinska and to the SciLife lab. And so the area that we study is, we try to understand human immune systems in general. And as a clinician, I treat patients, particularly children, whose immune systems behave in ways that we are not happy with, that cause disease because of dysregulation in the system, or because they have some inherent inborn error and so on. And so one of the problems that sort of has been for a while is that, as in many sciences, we've tried to reduce complexity to study specific processes, right? And that has led, in the field of immunology, where we study the immune system, to reduce complexity has been to resort to laboratory mice, where we can carefully control the genetic makeup of these mice. We can control their environment into a nearly sterile, actually in some cases fully sterile environment. And we can control every process, which has been incredibly informative. It's really led to incredible discoveries related to the immune system. The problem has been that we live in a very complex world as humans, with thousands, if not millions, of factors, microbes and pollutants and components in our world that that shape our immune systems, that shape the behaviors of our immune systems, because the immune system really is the sensory system for these environmental factors. And so how do we then learn something in a very reductionist system and then translate it into understanding of these complex systems in humans? That's really challenging. And so a complementary approach has been to try to study humans directly and then use sort of mouse models and reductionist systems to confirm the hypotheses that we generate in humans. And so that sort of new human immunology type of thinking is sort of a decade old and is in a field that we work quite carefully and heavily in. Now, another complexity with the immune system is that it consists of many different white blood cells. And they are specialized at doing certain tasks, which allows the system as a whole to do more complex things. But the problem is we cannot understand the entire system if we only look at one of the players one at a time. Again, this reductionist approach becomes challenging to translate into an understanding of the entire system. And that's where systems immunology comes into play, because what we're now trying to do with new technologies that allow us to measure all of the players at the same time from a given blood sample is to try to understand how these different factors communicate with one another and collectively give rise to behaviors, immune responses, and so on. And this is particularly valuable if we follow an individual over time, as we can do with human patients, especially if we look at blood samples. We can infer relationships between these different factors by following the immune system over time. So I wanted to give you a few examples of how we've taken some of these new technologies and addressing which I think is very important questions in human patients. So one of the things we've done is we've been interested in looking at very young babies, because this environmental exposure that I mentioned, it starts sometime right after birth, right? When the baby comes out, they're seeded by microbes, the microbiome is formed, and that early event needs to be studied, and we haven't really understood that. So to try to do that, we started setting out ways of how can we take a very, very small amount of blood, basically a drop of blood, and learn as much as we can about all the white blood cells in there and sort of. So that led to a lot of technological advancements, and actually a company that we founded, Cytodelics, which spun out to sell this to researchers all over the world, which allows us now to study the immune system in very small blood samples. So we could address this question of what happens when a child goes from a very protected environment in utero to the world outside, which is full of all these components that we all know. So we did that, and this is one of the reasons why this is important. This is epidemiological evidence from a number of studies showing that if you get antibiotics early in life, or if you have an infection early in life, or if you're born by cesarean section versus vaginal delivery, many of these diseases that happen much, much later in life, maybe even in middle age, are much more likely. Presumably because this normal colonization of the infant and the immune system sort of relationship there with the microbes is perturbed, okay? So that's one of the reasons why we wanted to study this. And from an evolutionary point of view, I think it's interesting to think about why do we have a sort of critical period early in life in humans where the immune system and the microbiome needs to get to know each other. Why aren't we allowing the system to be flexible throughout life? And I think one reason is probably because if we allow the system to be fully flexible throughout life, any infection that happens later on will throw everything off balance, and we would sort of lose the regulation of the immune system. So this is from a review that it's quite recently that I wrote about this critical period in humans. But basically, because we are sort of living in a sheltered environment, our mothers and fathers keep us close early in life. We get breast milk ideally, which protects the baby. We can allow a period during the first months of life where the immune system can get to know the microbes that colonize us in a safe sort of environment. And so we've studied this, and I'll skip some of these, but we've studied this process using a cohort gathered here in Stockholm at the Karolinska University Hospital, which is now about 400 infants that we profile longitudinally, both with blood samples and stool samples. The stool is quite easy to collect, as you can imagine. They're happy to share, but the blood is more tricky and it requires these very small blood samples. We've published a number of papers describing different features of this developmental path. And to summarize some of those findings in this slide, I basically wanted to say that we've learned that healthy microbes or beneficial microbes during the first months of life, they are characterized by being very good at metabolizing milk oligosaccharides, HMOs. They expand in breastfed infants. They tune the immune system away from a particular type of inflammatory response and towards a more healthy, beneficial response. They prevent inflammation in the gut in these babies. And all of this, I think, represents different aspects of this balance between microbes that are necessary for our health and the immune system that is sort of tolerating those microbes. So that's one interesting example. And I wanted to give you two more, the others a little bit shorter examples of how we're applying these tools to address questions that we think are important for human immune systems. And I think sex differences is another really interesting topic to me. And so together with colleagues Olle Kempe at Karolinska and Nils Landegren at Uppsala and colleagues in my lab and endocrinologists around the country, we teamed up to try to study sex differences in immunity. And we know that females typically have a much better outcome when infected. That's true even of newborns. So there's something throughout life. But on the other hand, females are typically overrepresented for most of the autoimmune diseases. So why is this sort of dichotomy here? On the other hand, males develop more cancers in general than females. And so there is something around, and COVID has been a perfect example of this sex difference, where here are a number of countries, and the case fatality rate on the x-axis on the left slide, showing that in all of these cases, individuals infected with SARS-CoV-2 have a higher mortality if they're men versus women. And that's true across the age span, more or less. So why is this? So to set out and understanding this, we teamed up with these colleagues that I mentioned earlier to do a study of individuals that undergo sex reassignment therapy. So these would be individuals that are born with either X and Y chromosome or two X chromosomes. And then they get hormone treatment over the course of 1 year, during which we draw blood samples to try to profile their immune systems in order to try to disentangle the effects of genes versus hormones. And the results are quite intriguing. The hormone levels, if we take this left example, which are females transitioning to males, I hope you understand what I mean by that. I don't want to be sort of condescending with the wording here. Females transitioning to men, reaching a male testosterone level after 3 months. And in their blood, we see some really interesting changes in specific white blood cells in purple that decrease or increase with time. I'm not going to go through the details. But the main take-home message from this study so far is that we have identified one particular white blood cell, which is quite rare, but very important in viral infections, also very important in specific diseases, called plasmacytoid dendritic cells. And we think they are sensors of sex hormones. And we think that they modify immune responses differently in males and females. I'll skip some of this for the sake of time. But basically, within the immune system, everything is about balance. And there are different factors that balance each other. And two of these are type 1 interferons on the left, which are the primary responses to viruses, and TNF-alpha, which is a pro-inflammatory cytokine on the right. And we think that these two are normally balancing each other out. And we think that what sex hormones do is they tip this balance, particularly via this cell type. All right, final example. I've been working quite a bit during this pandemic, initially with children who responded in strange ways to SARS-CoV-2 infection. As you all know, most children have very mild infections, fortunately, but some have strange presentations and some actually become terribly ill. But fortunately, we've worked together with colleagues, and I'm sort of Swedish coordinator for this global network called the Human Genetic Effort, COVID Human Genetic Effort, with partners all over the world, led by Jean-Laurent Casanova, one pediatrician and colleague at Rockefeller in the US. And what we've been meeting every Monday for 62 Mondays or something, and throughout this pandemic, and it's been incredible to be able to share data and insights with so many brilliant colleagues around the world. And we've started basically from my end by addressing this strange presentation that happened in some of the children that we started seeing in the spring of 2020, first in Italy, and then we've seen about 450 kids in Sweden now. These are children that come in to the hospital very, very ill, with high fevers for 5-6 days, these red eyes and rashes, and some of them are very critically ill. We haven't had any deaths in Sweden, fortunately, but internationally there's been several hundred children dying from this response, which is not directly caused by the virus. We now know that this is an immune response caused by the immune system, and the disease is called multisystem inflammatory syndrome, or MIS-C. And what we were able to do, thanks to this international collaboration, but also thanks to these technologies that we've been developing for profiling the immune system of children, we were able to do a first immunological investigation into this condition in 2020, which led to sort of an initial description that this is different from the disease Kawasaki disease, which we knew before. It is not the same as severe COVID, which we saw in adults. And I think this has also inspired some really impressive treatments now that we have for these patients. So I am incredibly grateful to a fantastic group of people. Everything that I've said is the work of these people, and also collaborators that I've mentioned along the way that help with various aspects of this work. Thank you very much.
Oh, Peter. Perfect timing also. So now I understand if I want to kind of improve my chances a little longer, I should change to become a woman.
It's not that simple. No.
Not that simple, all right.
But no, it's not that simple.
Not that simple.
I'm not going to elaborate on that.
Life is never simple.
Yeah.
Is there a more serious question? Yes.
Thank you for a wonderful talk, Petter.
Oh, hey, thanks.
So you say the Kawasaki disease and the MSC are different.
Yes.
Are there genetic differences? What's
Yes.
known about the genetic associations for the MSC?
That's a very interesting question. So Kawasaki disease, for those of you who are not pediatricians, Kawasaki disease was described in Japan, and it's 20 times more common in Japan than in Europe, for example. And we see a case every now and then after sort of flu season and so on in Sweden. However, the MSC patients have been nearly absent in Japan during the SARS-CoV-2 pandemic or COVID-19 pandemic. So these are completely distinct conditions. Actually, it was surprising that they had even fewer cases than we had in relation to the number of SARS-CoV-2 infections. So yeah, they're distinct when it comes to pathogenesis and everything.
Can I have one more quick?
Sure.
Did they have an increase in Kawasaki disease?
No. Actually a decrease, because some of these other viruses that we think trigger Kawasaki disease were lost, right, because of lockdowns and other measures.
Yes.
Thanks, Petter. So when you say that women do better when infected,
Yeah.
do you mean that in the sense that women clear the infection more efficiently, or that their immune system is not overactivating and causing a lot of other symptoms?
So that's a great question. I think the way I would put it is that we don't know if they clear the infection better, because some infections do become chronic, right? And so it's not like women are more likely to clear those infections. Think of EBV or CMV and so on. But when it comes to an acute infection in general, the outcome is better in females than in males. True even for newborns with bacterial, bloodstream infections and so on. And so it would mainly be that, the severity. And as in COVID, that's been very clearly illustrated, right? That the risk of dying if you're infected with SARS-CoV-2 is higher in males than in females. Now, the long COVID, this sort of persistent symptoms, seems to have the reverse pattern. And so,
Yes.
Yeah, and concerning this sex difference, this is really a complex situation, of course, and it relates back to how to address these things. And we were involved many years ago to look at epidemiology, given the best parameters to correlate rheumatoid arthritis with sex hormones.
right.
And was enormous studies doing this.
Yeah.
Outcome is zero. Very difficult to make conclusive evidence. It's too complex.
Are you talking
when
about
you come to the humans. If you go to biology, you can clearly see that actually estrogen protects against these kind of diseases. You could go. So it comes to this also. It's kind of philosophical also that looking at complexity of the system biology has its limitation.
Yeah.
It's you need to have a living organism to really address complexity. And most of what we know about complexity comes from mouse models, in fact. And then we translate it to humans.
Yeah.
This is two different directions. I don't think we should forget both directions.
And I agree with that. And I was trying to say that in the beginning, that I think some discoveries made in mice and other things can translate to humans and have led to revolutionary therapies. No doubt about that. But oftentimes we fool ourselves if we generate all of our hypotheses in a model system that is reduced in complexity. So what we try to do is to make observations in humans that then can be validated in a simpler system. There's no doubt that that's needed. We do that in vitro, in culture systems and so on. Absolutely. I think the one thing I would like to comment around the associations though is that if you're doing population studies, you will never be able to distinguish the effect of chromosomes versus hormones. And just looking at hormone fluctuations in relation to symptoms, I understand that that might not be sensitive enough. But in this case, the genome is fixed and the hormones change dramatically. And so in that system, I think we can see some differences.
Well, thank you.
Thanks.
I could just continue. Very interesting. Thanks, better. And now I'd like Ellen Moons to take the stage. Ellen is a professor in material physics at Karlstad University, and she got Foundation's Prize in Physics in 2011. Thank you.
Thank you, Christer, for this introduction. And I would like to start with thanking the Göran Gustafsson Foundation and Selection Committee as well for the prize. It has given me enormous possibilities, much beyond what I could expect. It has helped me to recruit PhD students, postdocs, build up a solar cell lab at Karlstad University, and also strengthened national and international collaborations in the field of organic photovoltaics and in emerging fields in solar energy. So that moment 11 years ago was very significant for my academic career. The energy from the sun that reaches the Earth, continents and oceans is about 5,000 times more than the global primary energy consumption, so all the energy we use on Earth. The numbers given here are in petawatt, that's 10 to the 15th watt, and terawatt, which is 10 to the 12th watt. The recent global primary energy consumption, GPEC, is about 16 terawatts. That corresponds to about 140,000 terawatt hours per year. That's what we consume globally, all of us together. And we have plenty of solar energy that comes to us, so there is no lack of energy. In 1 hour, Earth receives more energy from the sun than all the energy consumed by humans in a whole year. Among the renewable energy sources, solar energy has the largest capacity. The small cube in the right bottom corner is the global primary energy consumption, and the huge green cube represents the capacity of solar energy. So here it's a little bit more conservative than 5,000, it says 1,800 times, but it is still the largest one. Of course, fossil fuels have a huge capacity as well, but among the renewables, solar is really provides a lot of energy. We use that energy on Earth for many things. We convert it into chemical energy for plants, food. We use it as heat, but we also can convert it to electricity. And if we had the possibility to also transport and store that energy, there would be no lack of electricity anywhere. Among the sustainable development goals of the United Nations, number 7 here is the one that says that affordable and clean energy should be available for all. As a chosen symbol, the sun, and a kind of electrical symbol, it seems that it could indicate that solar energy is one of those energy forms that would be affordable, reliable, sustainable, and modern energy for all. At the moment, I think solar photovoltaics is a sustainable and modern energy form. If we can make it reliable and affordable, it will have a large uptake in society. And this is a picture from a seminar in this room, the Inevitable Transition to the Electrical Society, given by Professor Martin Green from Australia. And he showed us that today the installation of photovoltaics globally has reached 1 terawatt, and it has actually surpassed the installations of wind energy To highlight the wide range of possibilities of photovoltaics, I selected a few impressive projects. One is the Solar Impulse. It's the airplane that can fly without fuel. The only fuel is the light from the sun. This airplane has flown around the world in 5 consecutive days and nights without any additional fuel. And the other one is the solar tiles, which is a product of Tesla, where the light is converted to electricity and charges a battery with which you then can drive your electrical car. And the last one is the solar sails. So for all these means of transportation, solar could be used. And in the sail, flexible solar cells have been integrated in the cloth to supply power, not for the sailing, of course, but for electricity on board, perhaps. So what do you need to convert light to electricity? You need a semiconductor that is a piece of material that has a band gap. That can lead to absorption of light and generation of charges that can be taken out of this material. You also need to give these charges a direction, and for this you need a so-called p-n junction, which is a border between 2 different types of semiconductor. You need selective contacts to collect the charges. And the power conversion efficiency that will tell you how good the solar cell is, it is the ratio between the electrical power that is put out of the solar cell divided by the power of the light that comes in. How efficient are then these solar cells nowadays? Well, there are many different technologies available. The most common one is silicon. The most expensive one is gallium arsenide. The emerging one, the rising star, is perovskite. And the organic solar cells are the flexible ones and the ones I will talk about. You can see some numbers here for these power conversion efficiencies, and the best record cells in the world, they convert about 1/4 of the incoming sunlight's energy to electricity. There is a maximum limit stated by the Shockley-Queisser limit, which is around 30–32%, with a maximum where the band gap of the material is about 1.2 electron volts. Doesn't say so much, but several of these materials on the list have different band gaps than that. The higher numbers above this 30% cannot be obtained by a single p-n junction, a single solar cell. You need to stack solar cells on top of each other, where each of the cells would take their share of the solar light, of the solar sun spectrum, and convert it into electricity, and in this way help each other to reach higher numbers, like above 40%. These 2 emerging technologies, perovskite solar cells and organic solar cells, are areas with a lot of research also in Sweden. So the organic solar cells, they are consisting of a conducting ink, including molecules that form molecular semiconductors, where you usually use an electron donor molecule and an electron acceptor molecule. One of these are usually polymers, and the other can be a molecular polymer. They're mixed in a solvent, and that mixture will be your ink, which you then coat onto a substrate, and you can do that on a glass substrate, but also on a flexible substrate by, for instance, a printing machine. Here there are some examples, and the right top corner is a product by a Swedish company called Epishine in Linköping. Perovskite solar cells, they are either inorganic or hybrid between inorganic and organic, and they have a special crystal structure called perovskite they have a stoichiometry that could be assigned, that could be given as ABX3, where A is a monovalent cation, B is a metal cation, usually lead, and X is a halide anion. And those materials have given solar cells with very fast increasing efficiencies. Oxford PV is one of the companies commercializing that. So as I mentioned, OPV is a research field that is rising, that is growing very fast. The original works were done in the 70s, where organic semiconductors were giving photoconductivity properties as a single material, but with very low efficiencies. And only in '86, the first time materials were combined, donor and acceptor materials, where still the efficiencies are low, and only in the 90s efficiency started to grow in this field. So now we are working with these bulk heterojunctions where donor and acceptor molecules are mixed together and coated in one cone. As you can see, the Göran Gustafsson prizes in this area and related areas have been of significant importance for the growth of this OPV research field. That the number of publications is going down after a while, I think has to do, at least partly, with the rise of the perovskite solar cells. So Epishine is a Swedish company that is now building a factory that will employ about 50 persons. In OPV, they are producing light cells that can power sensors with indoor light. Arbor, PV Infinity, and Heol are also European companies in this area. In Karlstad University, we are working with several research topics. One is the morphology of these thin films of donor and acceptors, to control them and to analyze them. Another one is the degradation of these materials and try to mitigate that in order to improve the lifetime of the solar cells. And a third one is the interfaces with the neighboring layers. So let me tell you how such a cell looks like. So the cell consists of a photoactive layer that is sandwiched between two electrodes, a transparent one and a top electrode that is usually metal. And that is cast from solution, and in that process, when the film is drying, when the solution is drying, the solvent is removed and the donor and acceptor molecule start to phase separate. It kind of like when you have a sauce that phase separates, they form domains which are rich in the donor or in the acceptor. That kind of morphology can be beneficial or not beneficial for the solar cell, because as the light is absorbed and generates charges, the charges have to be able to reach the electrode, and that's a process with many steps where the morphology plays an important role. Early on, this fullerene derivative has been a very popular electron acceptor material. And recently, this fullerene was replaced by other electron acceptors. And with the help of that, the field has reached very high efficiencies, up to 90% power conversion efficiency. So from where we were a few years ago with about 10%, this technology has now become competitive with many of the other technologies in photovoltaics. These new acceptors, they absorb stronger and they have smaller losses, they lead to smaller losses, but they also aggregate more. And as I said, morphology is what we focus on at Karlstad University, and we use several types of tools like atomic force microscopy, transmission electron microscopy, and X-ray based microscopies like this STXM, which is scanning transmission electron X-ray microscopy, in order to find out where the donor and the acceptor sit in that structure and how we can improve the structures. This is a very large structure that leads to a bad solar cell, but even for smaller structures, we would like to be able to analyze the morphology. And the origin of that is thermodynamics and kinetics of this mixture. The new acceptors, they form much finer structures and are also more difficult to analyze, but they form this fine structure is part of the reason why the solar cells are more efficient. In this case, the strategy is to balance crystallization of this acceptor, so this kind of stacking of the molecule, because it's a planar structure that likes to stack, with the liquid-liquid phase separation that occurs if we have when there's no crystallization. So for this I want to mention one tool that we use, and this is a combination of an atomic force microscope and an infrared spectrometer, and where we use the advantage of the high resolution of the scanning probe microscopy and a very chemical specificity of IR spectroscopy in order to find out locally what composition we have under the tip. And this is one example of such a measurement. So you remember the big blobs that we saw on the left-hand side that we saw before, which are domains that are rich in this fullerene. Now, with this infrared technique, the two other pictures, the colorful pictures, we can now see where the fullerene is sitting, this middle picture, where we have yellow, that's where the fullerene is, and the right-hand side picture, this is where the polymer is sitting. So we can see the internal structure of these blobs. And even for the new acceptors, we see that we have a fiber-like structure surrounding the donor polymers. And the spectra we get are chemically very specific. So all this helps us in order to understand both the structure but also the relation to the properties. And we can conclude that the new materials that are coming now, they are driving the strong increase in power conversion efficiency of these organic solar cells towards 20%. The phase separation and aggregation, the balance between the two, determines the film morphology, and that also determines the properties. And smart engineering of this film morphology improves the device performance, and the operational lifetime is another thing we study, which I haven't had time to talk about. It needs to be improved. For flexible devices, it's months to years in the best case, and there's now publication out that for encapsulated organic solar cells, we could reach 30 years. So strategies are needed to stabilize the morphology as well as the photodegradation of the materials, and for this more research is to be done. So I think affordable, reliable, sustainable, and modern energy is there for all, within reach. Future directions could be green solvents, could be tandem cells, and more stable morphologies. One of these ideas could be to make a solar paint that you can use to paint your house or your car, and you immediately generate electricity. There's some work in Australia going on for that. With this, I would like to thank my group at Karlstad University and the funders, and you for your attention. Thank you.
Thank you, Ellen. Thank you for pointing out that the foundation has supported this in many times, more than I knew. So, a question to Ellen? Yeah, I think you were fastest.
This is obviously very important and currently of current importance for humanity, I can see. Now, in your introduction, you said that actually the total amount of solar energy is 5,000-fold higher than the consumption. What I want to ask is what do you include in consumption? Is that what's practically used by mankind? Or is it what's used by life in total, like, photosynthesis? What do you mean by consumption?
Yeah, it's this primary energy consumption, so I think it includes all consumption of energy.
Okay, so not just by mankind, but by life in general, or
Oh, I'm unsure about that. I think it's probably by
Because
mankind.
we can't use all of it, because then we are alone here.
That is right, that's right, yeah.
You said you wrote that these encapsulated cells, that they had 30 years of lifetime. Do you consider that enough? Is that enough for practical application?
Mhm. So this is a recent article from last year, and 30 years is a very good number, I think, because if you buy a silicon solar panel today, they will usually be sold with a guarantee of 20 or often 25 years of functioning. So 30 years is definitely, it's a surprising long time, but they are encapsulated in glass on both sides, so that's necessary, because if you have flexible solar cells that have plastic, there will be interpenetration of oxygen and water vapor, and that will degrade the cell together with light. So for most applications, you don't need even 30 years. But of course, if you think about putting that on your roof, you don't want to change them too often.
That's a quick question.
Can you say something about the temperature dependence of the PC? The temperature.
Yes, I can say something about that. So silicon solar cells have the nature that when you put them in a warm country, the efficiency goes down. Some of the emerging technologies don't have that drawback. So, but of course, there may be other things happening. For instance, if you have a molecular system and you increase the temperature, this automatically happens if you put it in the sun, of course, then these molecules may diffuse. So it's very important that this, the structure that you form that is giving you the high efficiency also stays there, and you don't get a degradation of your performance because of a temperature-induced mobility of the material. So it's a very good question. And this is something a lot of research is going on today about in all these technologies about the stability.
Well, thank you. I look forward next time I'm going to repaint my house. I know new paint I'm going to use. Thank you. And last speaker before the break, and that's paneldiskussion. Also a professor in physics at Chalmers, and he got a prize in physics in '99. Welcome.
Thank you very much. Actually, just like the first speaker, Pernilla, I'm born in Umeå and I ended up at Chalmers. So we have that in common, I realize. Here, I want to talk about superconducting circuits from artificial atoms to quantum computers. And I will, of course, on the way, explain what I mean by this. I'll start by a personal comment on why 1999 was an important year for me, apart from receiving the Göran Gustafsson Prize. Then I'll tell you about superconducting circuits, how you can build both artificial atoms and the basic building blocks of a quantum computer, namely qubits, out of these circuits. And I will tell you a specific experiment where we used one of these artificial atoms and listened to the sound of an atom. Do you know what the sound of an atom is? Have you heard the sound of an atom before? If you stay awake, you will have heard that when I'm finished. And then at the end, I will say a few comments about quantum computing, which is the project that I'm really working on now. And you see some pictures here on a cooling machine, dilution refrigerator, where we cool down these superconducting circuits, and this is a chip with 5 of these qubits. So first, why was 1999 a special year for me? Well, 1999 was the year when the first superconducting qubit was demonstrated by a Japanese group. That got me very interested because they used exactly the same technology that I had been doing research on for 15 years. And I realized that this is something where I could use my research to do something really interesting. So, And that then coincided with me getting the Göran Gustafsson Prize in Physics. So it gave me money to employ several people to work on these things. And I sat down and thought about what can we do, and what can we work on, and how can we make an impact. So of course I'm very grateful for this opportunity that I had this chance, that this coincidence actually happened in the same year. So we started working on that, and a few years later, in 2003, published in 2004, we managed to do our first superconducting qubit, which you see. I think this is maybe not visible on the screen. But you see here on the picture a superconducting qubit, which was very similar to the one that the Japanese had done, but we had added a way to read it out, which was, in my view, more clever and nice than what they had done. So then, what is a superconducting qubit? So first, superconductivity is when a metal loses all electrical resistance. So in other words, the metal doesn't get warm when you run current through it. That's a very nice property if you want to build something that should be quantum mechanical. And so what is a qubit? That is the basic building block of a quantum computer. And it's actually possible to understand in simple terms what it is. So here you have a circuit which has a capacitor and it has an inductor. So these two plates here, you can store electric charge on those plates. So you store the energy in the electric field between those two plates. This charge can go through the inductor, creating a magnetic field. So you change the energy to the magnetic field, and then the charge goes back and forth, typically 5 billion times per second. So you have a frequency of 5 gigahertz, and the energy is going between the electric field and the magnetic field all the time. So that's an oscillator. And the energy spectrum for that looks like this. You have equidistant levels. So whenever you put in energy, you have to supply precisely the right energy to go from, say, 0 to 1 or 1 to 2. So this is essentially counting the number of excitations in the circuit. However, this is not enough to make a qubit or an artificial atom. So what you need to do is you need to include a nonlinear element in this circuit, and what you will replace is the inductor. So there you put something called a Josephson junction, which acts as a nonlinear inductor, and that changes the energy spectrum of this oscillator so that it becomes anharmonic. So you see that it's a different energy between 0 and 1 and between 1 and 2, and that is precisely like the energy structure of an atom. So in that sense, we can call this an artificial atom. And this allows us to tailor-make artificial atoms. And we can, for this reason, study effects that are not possible to study if we use natural atoms from the periodic table. So it gives us a way to tailor-make these things. So what do they look like? Well, they are made with nanolithography on a silicon chip. So the dark here is silicon and the brighter is aluminum. And you see in the middle here, there's a small circuit that is too small to see in this picture, which is the actual Josephson junction that gives this nonlinearity that gives you all these properties. So now, if you, so this is then an artificial atom, and I will talk more about that, how we use that. But you can then also concentrate on only the two lowest levels, and then you can encode information in 0 and 1, and then this can be used as the building block for a quantum computer. So I'll tell you about both of these aspects of this kind of circuit. But it's the same circuit in both cases. So first, one more thing I wanted to say. So if you excite this artificial atom from 0 to 1, and let it just sit, it's going to decay after some time. And usually it will send out a quanta of light, so a photon. So this is what we're used to in practically all situations. But we were interested, can you also send out something else? Can you send out sound? And we found a way how you can actually make this artificial atom more strongly coupled to sound than to light, so that it emits a quanta of sound, namely a phonon instead of a photon, when it decays. So how did we do that? Well, what we used were so-called surface acoustic waves. Those are like ripples on a surface like this, so you can think of water ripples on a pond, but this is on a solid surface instead. So these waves, they are called Rayleigh waves, is the same kind of waves that you have in an earthquake. They can travel a long distance and can be felt at a very long distance from the epicenter. So now how can you then make these surface acoustic waves? Well, you can do that using a piezoelectric surface. A piezoelectric material is something where you apply a voltage and that changes the size of the crystal or the substrate. And especially what we use is this kind of interdigital transducer where you have fingers like this. And you put a voltage between those two fingers. So you get an electric field in the substrate, and that then changes the distance. So it compresses or increases the distance between the fingers just because you put on a voltage. And so if you put on many of those together, you can get a sizable effect where you put on a voltage, and then you have a wave that goes out in both directions from this transducer, what I call the IDT. You can also do it the other way around. You can send in a wave, a surface acoustic wave from the side, and that is then picked up and converted so that you convert it to the electromagnetic domain. So in a way you can say you convert phonons to photons and photons to phonons. So it's a converter between sound and microwaves. So that we can use. Now the next thing to realize is that this superconducting circuit, which I call the qubit or artificial atom, actually looks very much like the IDT that I just showed you how to do a surface acoustic waves with. And so what we did was we took this kind of superconducting circuit and put it on a piezoelectric surface. And that allowed us then to couple the artificial atom to sound. And this coupling was made so strong that it was much stronger than any coupling to light. And so that allowed us then to make experiments with sending surface acoustic waves to this qubit or artificial atom and making understanding the physics of this. And so we so what's important to understand here is that this artificial atom actually has two important frequencies. One is the frequency at which it stores its energy. But the other frequency is set by this finger distance here, and that is the frequency at which it talks to sound. And moreover, since we have, if we make here two Josephson junctions in parallel, we get a tunable Josephson junction, so we can tune the frequency. So we can tune this frequency where the atom stores its energy in and out of the frequency where it talks to sound. So we have control over when it couples to sound and not. Now, so we made several experiments. One is where we sent, we generated surface acoustic waves and sent it to the atom. So acoustic in, acoustic out. We also listened to the atom. So we had a separate control line, the red one, where we excited the atom, and then we just listened to when it emitted sound. That's what I will show. So that's electric in, acoustic out. And then we also did both things at the same time. But the only thing I will have time to show you is the middle one when we listened to the atom. So here you see also the people who did this experiment. Mainly Martin Gustafsson was the main person, and Thomas Aref was the one who recorded the sound that you will hear in a minute. So what we did was we had this gate here. So on the top of the blue, you see one excitation there. that we use to excite the qubit. And there's a little bit of crosstalk. So and this is our detector here on the other side. And then we amplify it and send it to the loudspeaker. So first, when we send in a signal, you're going to have some crosstalk. So you hear our excitation signal. But then, at 40 nanoseconds later, you will actually listen to the sound that comes to our detector. And that's the sound of an atom. Then there's also a finite probability that the sound will actually bounce on the detector, bounce on the atom and come back again. And then you will hear an echo. That's the third sound. So first you will hear the excitation, then the atom sound, and then the echo of the atom sound. So listen now. This is the excitation. Now comes the sound of the atom. And there comes the echo. I'll do it one more time. Sorry. Excitation, atom, echo. Now, of course, we have fiddled with this sound to allow you to hear it in two ways. One, the sound here that came out was 5 gigahertz. And of course, we had to reduce the frequency so that you could hear it. Also, the whole trace here took 200 nanoseconds. You wouldn't be able to hear that either. So we just extended the time. But doing those two changes, you can actually hear the sound of an atom. Good. Now we come to quantum computing, because as I said, we can use this superconducting circuit not only to make an artificial atom, we can also make a superconducting qubit. So we can encode information in this circuit. And just to compare, in a classical computer, you have bits that are represented by a voltage state of a silicon transistor, typically. So this is made with semiconductors. But you can only represent 0 or 1. You have to choose one of them. In this quantum computer where you have qubits, the special thing about quantum physics is that you can have superpositions. You can represent 0 and 1 in the same bit, in the qubit. And so you can have both 0 and 1 simultaneously. And that is really what it makes this difference, that this behaves according to quantum physics, this behaves according to classical physics. And I'll show you why that is important. Superposition. So let's take an example. So the qubit, so you can think of the qubit as this artificial atom. There you can encode, in one qubit you can encode 2 numbers, 0 and 1. If you have 2 qubits, you can encode 0, 0, 0, 1, 1, 0, and 1, 1. So 4 different numbers. And then you can consider how many numbers can I encode if I just add on more qubits. And it turns out it doubles every time you add a new qubit. So if you have, say, 4 qubits, you can do 16 different numbers and so on. So this scales as 2 to the n, exponentially with the number of qubits. So it's actually enough to have only 300 qubits. That's 2 to the 300, which is approximately 10 to the 100. And then 10 to the 100, that is much more than there are particles in the universe. So that means with 300 such qubits, I can encode so many numbers at the same time. And these numbers are more than the particles in the universe. This is the magic of quantum computing. And this is what we want to harvest, trying to build a quantum computer. Now, another way to display this is this analogy with there's this anecdote of the person who invented chess and asked for a small prize from the king for having invented this nice game, where he asked, oh, I will only ask for one grain of rice for the first square on the chessboard, and then 2 on the next one, and 4 on the next one. And the king said, oh, of course, that you'll get that. Now, he did not understand the concept of exponential growth because you see that when you come to square number 64, that corresponds to more rice than is produced every year, even today. And of course, the king in those days did not have that much rice to give. So he had to do something else. Yes. Okay, so I don't have time to explain all the things we're doing today, but we're trying to build a quantum computer within this Wallenberg Center for Quantum Technology that I'm leading at Chalmers. So we have essentially a 12-year program, and we have two tasks. One is to build a Swedish quantum computer, and one is to enhance quantum technology knowledge in Sweden, both in academia and industry. And this is what I spent practically all of my time doing today. So, and presently we are building a 25-qubit processor. Each of the corners in the red matrix there is a qubit, and the other layers are different connections. We cool that down to very low temperatures, 10 millikelvin. And I should also say something about what can you do with the quantum computer once you have built one, which is, by the way, a research topic on its own, because we don't have a quantum computer, so we don't know what is the best use of it. But a few things that you can do is optimization problems. For instance, logistics optimization is a good task for a quantum computer. It can also be used to simulate complicated things like large molecules and new materials. It can also be used for code breaking. You can break the codes that we use on internet. That's possibly not a good thing. But I also have a dream that we should be able to break Bitcoin, and not for earning the money, but for preventing other people from using a lot of energy to mine Bitcoins. And then if we succeed in that, we will spend the money we earn on the Bitcoin to build a bigger quantum computer. Thank you for your attention, and thank you. And I want to thank the Göran Gustafsson Foundation for really coming in at the right time in my career and really helping me to boost this kind of science. Thank you.
Well, thank you, Per. I'm very glad that we could come in at the right time. And as a physicist loving fundamental physics, this quantum thing is really exciting for me. But we have time for one question, I think, before if there's a question. Yes.
Yeah, sometimes we hear on the news that someone has built a quantum computer that's calculated something, and someone else says that's not a quantum computer. So is that what's going on there? You know, how can we not know, be sure, whether it's quantum computing or not?
In many cases, this is done by companies that sometimes oversell what they do. And the criticism often comes from other companies who are also trying to build a quantum computer. So there's some psychology to that. But there are quantum computers. They're not yet so powerful so that they can do something that a classical computer cannot do. That is actually the next step. And we do know that quantum computers can do things that classical computers cannot do. And that has been done, but not something useful. So the next step is to do something useful.
Well, thank you. So, for everyone inside here, there's now fika outside here for you on the wherever you are on the internet. I hope you have a fika close by, and back here at 3 o'clock sharp start. Without bringing in anything. I'm back in time. Also hope you on the streaming side. So 4 more exciting presentations of our science prize winners, and then we will have a panel debates. So first of all, now we'll have Torkel Klingberg, professor in cognitive neuroscience at Karolinska Institute, who got our prize in medicine 2011. Please take stage. Very welcome.
Thank you. Thank you very much for the invitation. I'm honored to be here. If you're feeling tired, I would like to offer you some cognitive workout here in the afternoon. So if you look to the left there, you just remember where this person is pointing so that you can point at the same boxes in the same order. Okay? So what you're doing right now is you're keeping 4 items or spatial locations in your working memory so that you can point at the same boxes in the same order, right? Not too difficult. If you're feeling too good about yourself now, I'm going to give you another test. Any volunteers here? So that's more difficult. And the reason is that your working memory capacity is limited. And by using tasks such as this one, where you gradually make it more and more difficult, increase the information load, you can get an exact number on your capacity limit, because that differs between individuals. And surprisingly enough, this capacity limit is predictive of a lot of important stuff. It is highly correlated, one of the most highly correlated tasks to fluid intelligence. It correlates to performance in school, but also the trajectory of learning. And its working memory deficits is a core cognitive deficits in ADHD related to inattention. So the big question for me and for others have been, what is the cause of this capacity limitation? And the answer might be found in the brain. And people started to dig into the brain in the 70s and 80s with neurophysiological recordings. And it's easy tasks where a monkey is retaining just one spatial position in working memory. And then you can record the neural activity during the delay when the memory is maintained. And unlike long-term memory, where you store something by, for example, modulating synapses, this is not a structural remodulation of the brain, but it's electrically coded. So information is coded by this increased delay activity when you're keeping information in memory. And a lot of these studies led people to actually create computer models of how this information is stored, that I will show later. But these neurons can be found then both in the prefrontal cortex and in the parietal cortex of macaque monkeys. And how is the information labeled? Well, it looks like it's different neurons that is coding for different positions. So it's kind of a map that you can actually have in the brain. And biophysically realistic computer simulations suggest that we have recurrent networks here. It's kind of a positive feedback loop that is keeping this information in mind. So the bump of activity here is the equivalent of the memory in the brain of this position in this case. And what people have found in monkeys and in computer models seems pretty consistent with what we see in human neuroimaging too. So during delay, when you're keeping visual spatial information in mind, for example, we see sustained activity in the parietal and prefrontal cortex. And it also seems to be a kind of a map. And it's actually so precise that by recording brain activity when you're keeping information in mind. We can predict what kind of information that you're keeping in mind. So if you remember something up to the left corner, we can see that from the scanner. One question that was discussed early in my career was to what extent it's totally parallel networks or multimodal areas too involved in working memory. And our research and others suggests that there is both, but there are kind of a key network, frontoparietal network, that is involved in different types of working memory. So independently of you, if you're remembering positions here, or if you're remembering digits, for example. So what does that mean? You could draw two conclusions. One is that if you perform these two tasks at the same time, they would interfere with each other because they demand activation of the same networks. So for a couple of years, I and my colleagues performed multitasking experiments. But the other conclusion is if you could somehow enhance the performance or functionality of this network, for example, by training this task, you would benefit also in other tasks. So that was the next thing we tried. So I teamed up with computer game programmers to do a kind of a game-like training here, which looks like this. And we tested this much the same way that you do with pharmacological studies, so randomized, controlled, placebo-controlled trials, and found that yes, we could improve performance, and the performance was also enhanced for tasks that were not specifically trained. So we have a transfer. So following this, there has been a wide range of studies looking at not only specifically the type of method that we used, but it's been hundreds of studies, other types of working memory training. And here is results from 6 meta-analyses of different working memory training trials, suggesting, yeah, you can, compared to a control group, you can improve your capacity around 0.6 standard deviations. It's not a silver bullet, but it takes someone with deficits into the normal range. Another question was, to what extent does this affect inattention? And in one of our first studies, we did see that kids with ADHD decreased symptoms of inattention in everyday life, which has to do with the link between working memory and attention. And this also seems to hold up, not only in ADHD, but also in some other patient groups. For example, kids that are treated for cancer, where cancer affects the brain development and they get working memory and attention deficits. We can help them with this training. What about the neural basis? Well, one of the early studies suggested that when we do this training and relate changes in brain activity with changes in capacity, we see changes in this key network that we identified from the start, this frontoparietal network. And this has been shown then with a lot of neuroimaging techniques by others too. One question is, of course, what is happening in these regions? And that's not so easy to see in humans here, but other researchers have done studies with monkeys doing working memory training and showing that after training, you can see a larger percentage of neurons engaged in maintenance. So this is a plasticity at the cellular level, a specialization that we see. And also we can see an increase in the frequency, the firing rate during this delay, which would correspond then to what we see with MR scanners, for example. Another theme of plasticity is this with connectivity. So it's just not one region, but it's a network involved. And the connectivity between regions is probably important. and people have shown that it is effective also in working memory training. So one way to look at that is to just look at how is the correlation of brain activity in different brain regions. And if you can see increases in this correlation between, for example, the frontal and parietal region. Another more invasive approach is what we're looking at here. What they used is a magnetic pulse to the parietal cortex here, and then they record the reaction in the rest of the brain. And then they did this before and after working memory training, and found that you see an increased connectivity. So you have a larger reaction, which is an indication of a stronger connectivity. Yet another dimension in plasticity here is dopamine. So it's known since before from neurophysiology and from human studies that working memory is dependent on dopamine. And this is not a linear relationship, so it's not that more is better, but it seems to be an optimal level, and you shouldn't have too little or too much. So we were interested in looking further into this. So we looked at young healthy volunteers. They did psychological testing before and after training. We scanned their brains to find the working memory regions of these persons. And then we looked at receptor density of D1 and D2 receptors, and found that we have a model, again, this non-linear model showing a relationship between the baseline density, improvement after training, and the density afterwards. So it's a kind of a fine-tuning of the dopamine transmission here. So the interest in dopamine has also led to some genetic studies where people look at candidate genes to see if they involved in working memory training somehow. And we were also doing some of these studies and finding some indications of genetic involvement. Now this approach has kind of been abandoned now, because people see that even if you can find individual genes, they often explain very little of the variance, and it's often difficult to replicate these studies. And another approach is, instead of looking at many places in the genome, and this is called polygenetic score. So what you do is you're looking at thousands of places in the DNA, and you optimize the weighing in of these for something in order to predict something. And the something that people have been most successful in predicting is educational attainment. So how many years you spend on education? Is it just high school, or is it a master, or whatever? And Lee here used this method to in more than 1 million persons and got a polygenetic score which was more powerful than anyone had found before. And it turns out that this score is also the most predictive for IQ. So we thought that this would be interesting to look at. So we looked at two phenomena, working memory development and working memory training. So when kids get older, they get a better working memory. We don't really know why. When they train, they also improve. And the second graph here shows that there is a variability. So some improve more or less after training, and they develop faster or slower. And then we looked at this polygenetic score to see if it had any relationship. And it turns out that it is significantly correlated to both phenomena here, with no significant difference between the associations, so no interaction here. So this is pretty interesting. So it means that development could be seen as kind of a plasticity thing. That is, it's not a hardwired thing, but the development we see normally might partly be driven by challenges in everyday life. So to summarize here, there are now many studies in mice, in monkeys, in humans with different techniques suggesting that we have changes in functional connectivity between frontal and parietal cortex. We have modulation of frontal and parietal activity. We have changes in striatal dopamine release. Some of that might have to do with dopamine, gene-dependent differences in dopamine signaling. The neurophysiology shows that there is firing rate during the delay that is changed in neurons with delay activity, and computer models suggest that there is this could be strengthened intra and inter network connectivity underlying this. One question that I posed in the beginning was this relationship to school performance, and this has been kind of an unclear point for a long time, but it's now just the last 2-3 years it's been clearing up. So there's been a lot of studies looking at math performance before and after working memory training. Some finding effects, some don't finding effects. Most of the differences could be due just to statistical noise and the fact that most studies are simply statistically underpowered here. But if we look at the two largest ones, there is one negative study in 450 kids with low working memory. But then more recently, there is a German study looking at almost 600 children, again, training or control, and showing that you do have an effect on working memory and mathematics too. It is small directly after and then gradually comes. So when they follow up after 6 months and 1 year, and actually after 3 years after training, they had higher chance of entering academic track or Hauptschule. So this is interesting. Could it be because it's differences in the populations here and that some populations are more easy to train than others? So we tried to settle this by having a really large study. So we entered 17,000 kids and they were randomized to having different amounts of working memory training, and they were 6 to 8 year olds. Now we could see that they do improve. The improvement is related to how much working memory training they do, but it's also related to the baseline working memory performance. So this points to individual characteristics, and with the genetic knowledge now, we can hypothesize that it's probably the same genetic underlying both baseline performance and also improvement from training. So in the end, working memory training might not only be interesting to study, but it might actually be pretty useful too. Thank you for your attention.
Thank you. I almost wished I could improve my memory. There's still no fast way to do it. Right? Question? It's time for 1 or 2 questions for sure. Yes, Arne.
Okay, you mentioned a lot of studies on kids or individuals up to 25 years of age. Is there any hope for Christer to improve his memory with
Yeah, I wanted to ask that actually.
Or, I mean, if you go up in age.
Yeah, it's been done. It's been done in like healthy, young and older volunteers, so up to 65, 70 years of age, without any diagnosis or so. They improve less than 25-year-olds, but they still improve, and you still see an effect on everyday behavior, like attention in everyday.
Not too late.
Not too late.
These tools to improve the working memory, where do you find them?
I'll show you later.
Right, last question.
Yeah, it was exactly the same question. How hard is it? Like, do you have to practice an hour a day or?
Yeah, I didn't go into the details. It is hard work. It's been a lot of different approaches to this. Some experimental, some driven by companies. I have association to one such company, but it is demanding. It takes around 45 minutes per day for 5 weeks, and it's extremely boring.
All right, go home and work tomorrow. No, thank you very much, Torsten. And like Anders Johansson. professor in astronomy at the Lund University, also at the Copenhagen. We got the prize in physics in 2019. Welcome.
Thank you. Thank you very much. It was a great honor to receive the prize in 2019. It's a great honor to be here today also. And today I will talk about the work that we do in my group on forming Earth and also other planets. You see another planet there. And my groups are located both at Lund University and now more at the University of Copenhagen. Yeah. If we want to talk about other planets, let's start with our own planet, the Earth. So here's kind of what you can maybe call an astronaut view of our planet. Our planet is a terrestrial planet or a rocky planet, has a solid surface. It orbits the sun, which is a star. That's sort of the definition of being a planet. Earth has a radius of 6,000 and a bit of kilometers. So this globe there is like something like 12 or 13,000 kilometers across. Earth has a big mass, a 6 with 24 zeros after, and a big moon. One thing that's unique about the Earth in the solar system is that there's liquid water at the surface. Oceans. Not only have we oceans, so we are not fish, we also have continents, you see these continents there. And of course, Earth is very dynamical also. It looks a bit boring when we look at it like this. If we play the Earth movie over millions of years, you'll see the continents moving between each other. It's a very dynamical system because of this water and the plate tectonics. We have an atmosphere with oxygen, which was created by life, but also maintains life. And not only do we have water in the oceans, we also have rain and snow, so the water in the ocean gets washed over the continents and make the continents habitable also. And all of these properties of Earth means that life originated here 4.5 billion years ago, and that has been stable ever since for 4.5 billion years. So very impressive. If you look at our Earth in the solar system architecture, then we have to go here to the inner part of the solar system. Within the asteroid belt, you see a blow up here. We see that we have the 4 terrestrial planets. We have Mercury close to the Sun. Then we have Venus, and then we have Earth, and we have Mars that all have solid surfaces. Then we have the asteroid belt there with all these rocks. And then beyond the asteroid belt, we have the giant planets, both Jupiter and then Saturn, and then Uranus, and then Neptune as the outermost gas giant planet. The unit here is AU, astronomical units. That is illustrated here with the purple line here. That is the distance between the Earth and the Sun, about 150 million kilometers. All right. But today we live in a very interesting time where we know not only of the planets in the solar system, but also planets beyond the solar system. The first planet beyond the solar system, so-called exoplanet, was discovered in 1995. That was, of course, awarded the Nobel Prize in Physics in 2019 also, so another big prize handed out in 2019. And today, astronomers know of more than 5,000 exoplanets. I'm illustrating there and there on the top left plot. Along the x-axis, you have the distance from the star, but measured in the orbital period in days. So 365 days would be the Earth, and when we come closer to the star, we orbit faster also. So Mercury would be around 100 days. And on the y-axis, I'm showing you the size of the planet where Earth is 1. So 1 is Earth, 4 is Neptune, 10 is Jupiter. And there are lots of dots on this plot. Just wanted to show you how many planets we know around other stars. But they're all very close to the star. That's not because there are no planets farther away from the star, because they're much easier to find. With various techniques, it's much easier to find planets close to the star. Now, if you want a planet like Earth that has liquid water at the surface, we better be at the same temperature as Earth. And then we've got to move out beyond the left plot and go to the middle one. And now I have a different x-axis, and that is the irradiation that the planet receives relative to what Earth receives. So 1 would be the same surface temperature of Earth, and when we go to higher value, we get hotter. When we go to lower values, then we get colder. And we see on the top there, you see Earth and Mars. Earth sitting at 1 around a star of 6,000 Kelvin. That's what we put around the star. We have Earth in the habitable zone where water is liquid at the surface. We have Mars at the outer edge. Mars is frozen, no water, no liquid water there. And we see sort of a couple of handful of planets there that are orbiting in this habitable zone where water could be liquid at the surface. Most of them orbit though around stars that are colder than the Sun, 5,000, 4,000, 3,000 Kelvin. It's a really cold, small star that's called M dwarfs. One of them is written in the middle there, Proxima b. Proxima Centauri is the nearest star to the Earth, to the Sun in our galaxy. And a few years ago, a small planet was discovered around this M dwarf star. This is not a telescope image. This is an artist illustration of how this planet may look like. So there's a totally different kind of Earth. Why does it look this way? Possibly because it orbits so close to the host star that, like the Moon is orbitally locked to the Earth and always shows the same side to the Earth. This one is so close to the host star. It always shows the same side to the host star. So the front side is in eternal light and has just probably completely deserted. And the front and the back side is eternal darkness. So it's like a big Antarctica. And maybe there's a habitable strip of land in between. Just where you have the right temperature and the glaciers are melting into the hot side, and then it's probably sublimating and then moving over to the dark side again. So this is an example of a potentially habitable planet around another star. And what I'm interested in answering in my research is sort of how do such small potentially life-bearing planets form, and also where do they get their atmospheres from, because the atmosphere is key to understanding the origin of life. Okay, all right. But we can continue with the observations because we can go out now and look at stars that are giving birth to planets right now. We can find young stars that are only a few million years old. And when we look at them, they look something like this. And this is not the star. The scale here is about 100 astronomical units about the solar system. And what we're seeing here is a millimeter radiation observed here with the ALMA telescopes. It doesn't come from the star. It's a stellar light that heats a disk of gas and dust around this star. And it re-emits at thermal wavelengths, so 30, 40, 50, 100 Kelvin. That comes out in the millimeter wavelengths. Actually, these disks contain 99% gas, hydrogen and helium, which we can't see at these temperatures, completely transparent. They contain 1% dust and ice. This is what we're seeing here. And this dust and ice is what comes together to form planets. So we can observe planet formation right now. If you look at the very young stars, they will have several hundred Earth masses of dust. So that's more than enough to build our solar system. But if you look at stars that are older and older, a few million years old, after a few million years, they're losing all of the dust and also all the gas. So we know then that planet formation must occur in a few million years while they still exist. That's why I write here that planet formation is a race against time. Got to be fast. Okay. So one of the things I'm very interested in studying is the first step in planet formation. That is when micrometer-sized dust grains in this disk collide and stick together and end up forming something like these asteroids here. So the asteroids in the asteroid belt are so-called planetesimals. So they are left over from the formation of the solar system. And it's very interesting to study these planetesimals because these are the oldest objects in the solar system. They are older than the planets. Yeah. So we start with the dust grains, then they collide and stick together to something like millimeter, centimeter-sized pebbles, but they're very fragile. Their dust aggregates take such a pebble between two fingers and it will pulverize immediately. So they have very poor sticking properties, and sort of a mystery how to get from those pebbles onto these planetesimals, these asteroids here. So when I was a PhD student, I decided I wanted to develop a computer simulation and study how these pebbles move around in the gas in the protoplanetary disk. And I found something like this third slide here, third figure here, where the pebbles spontaneously come together and form sort of dense filaments, a little bit like migrating geese or bicycle riders in the Earth. There's sort of spontaneously aerodynamical instability that's called the streaming instability that makes these pebbles come together. And when they're coming together, at some point, they will come so close together that the gravity in big clouds of pebbles becomes so big that it sort of draws the pebbles together to form planetesimals like the one in the middle and the asteroid of 100 kilometers. So this is a way to go from millimeter-sized particles to 100 kilometer-sized particles without relying on sticking or collisions or anything like that. It's simply gravity pulling these things together. Yeah. So when we run these computer simulations, then we can form a number of little dots on the third panel here, and we see these are all planetesimals. Typical scale, 100 kilometers, very similar to the asteroid belt, so that's a pretty good test of the model. If the asteroids are the same size, that's a good test. We also form some smaller ones. And very excitingly, just occasionally, you can see with the lower green arrow points at two objects that are orbiting around each other. You may know about binary stars, two stars orbiting around each other. Here we have binary asteroids, two asteroids born orbiting around each other. And actually, if you go to the Kuiper belt, there's an asteroid belt that exists beyond the orbit of Neptune, the outer edge of the solar system. There are many, many binary planetesimals there. Okay. So this Kuiper belt had not been studied observationally or in situ until the New Horizons mission flew past Pluto in 2015. Pluto is not considered a planet anymore. It is the biggest object in the Kuiper belt. It's a big asteroid, so to say. And the New Horizons mission first flew by Pluto, that's on the top left there, and then it decided to find a smaller object, much, much smaller object. So the one below is like 1,000 times smaller than the Pluto. It's only something like 30 kilometers in the size. It's called Arrokoth, and when New Horizons flew by Arrokoth, it turned out that it was a whole new object that we've never seen before. It is not a binary, it's a contact binary. Two perfect spheres lying on top of each other. It probably used to be a binary, and for some reason these two components collided very gently and landed on top of each other. So there are many binary asteroids and contact binaries in the asteroid belt, and this supports planetesimal formation by the streaming stability mechanism that I explained before, because that's a good way to form many binaries. So when this news came out, then there was a reporter from BBC who called me suddenly up and said, "Are you aware that NASA is going to confirm your theory for planet formation?" And I said, "No, that's great." And he said, "How are you going to celebrate it tonight?" And I said, "We are going to have pizza and Coke, because that's what we always have in the family Friday night." And the reporter said, "Could you send me a picture?" I said, "Sure." So you can see on the top how NASA celebrate, hugs and high fives and so on. And this is how we celebrate in school, sort of eating a pizza and saying, "Oh, that's great." But it was a very good day when the reporter called and said that NASA had confirmed my PhD work. Yeah. All right. But beyond that, we still want to form planets, and we're not quite there yet. We're only at the 100-kilometer planetesimal stage. And traditionally, people say, fine, they're going to collide, all these planetesimals, they're going to form all these planets. But if you really do this in a computer simulation, it's not that easy. I'm showing sort of an illustration here of a planet in the middle that's trying to grow. It's creating a planetesimal, that's sort of the middle-sized star. And what happens, like in physics, when two objects with a force encounter each other, there's a scattering. Scattering doesn't mean collision, just means changing orbit. Replace the planetesimal by the small pebble, the small blue pebble, and the blue pebble is experiencing a lot of headwind from the gas, and on the way to passing by the protoplanet, it loses so much energy that it goes from being unbound, gravitationally far away from the protoplanet, to becoming bound. It loses energy, gets a negative total energy, and the pebble has no choice but to spiral and become accreted by the planet. This is what we call pebble accretion. That's something that I worked on since something like 2012, 2013. And pebbles are accreted much faster due to energy dissipation by gas friction. So the same pebbles that make planetesimals now continue to drive the growth of planetary systems. And when we run simulations now of how to form planets with pebble accretion, we get beautiful populations. Along the x-axis, we have the distance from the star in astronomical units, 0.1, 1, 10, 100. 1 was Earth. The mass in Earth mass is 0.1, 1, 10, 100, 1,000, 10,000. And you can see the 4 crosses out there that we wanted to match. The 4 pluses up there, these are Jupiter, Saturn, Uranus, and Neptune. And we can actually form them with pebble accretion. So goal accomplished. Now we understand how the giant planets form. But then look at Earth and Venus there, at 1 AU and 1 Earth mass, 2 pluses in the middle of the population. So we also form, as a side effect, Earth and Venus. But hey, that's something wrong, because people say they should form in a different way. Earth and Venus should form this way, according to the classical theory. Get a lot of planetesimals, grow into protoplanets, they collide over a few million years, and eventually you form something like Earth. It should take 30 to 100 million years. Yeah, because we don't really need the protoplanetary disk there. Even more importantly, you see this illustration of the giant impact here on Earth. The impact also delivers ice from an icy asteroid, and that becomes the ocean. So those are traditionally people would say all the life-essential volatiles, hydrogen, carbon, nitrogen, is delivered by a giant impact. That's completely random. Whether the Earth is dry or an ocean planet, it just depends on how many of these collisions that Earth encounters. Now, on the other hand, we're proposing this picture now, where the pebbles are raining down through the planet and eventually landing in the magma ocean. The planet is completely molten here. And those pebbles are carrying water, ice as rims. They're carrying little organic molecules, not the ones that make life, mostly C and N carry molecules that will be destroyed later. And then we need them later to form life again. And one thing that's nice about this mechanism, first of all, it takes only a few million years. Now the planet forms much faster, Earth forms much faster. Secondly, when the volatiles, the life-essential volatiles, are delivered by these pebbles, then it's completely calculatable how much HCNN that the Earth gets. It's not random anymore. It's like something that you can calculate. So the amount of hydrogen, carbon, and nitrogen that Earth has is now a consequence just of pebble accretion and pebble drift. And this also implies that there may be many planets out there have a similar amount of water and carbon as Earth has. It's not a random thing. This is maybe the most common thing for a planet that forms around where the Earth formed. And one thing I'm very interested in now in my research is to go on from sort of models of forming planets to think about what is the first atmosphere that becomes outgas from these planets, because the first atmosphere that comes out of a planet is the conditions under which life originates. And I want to understand how common life is in the universe. A funny thing, though, first, is that if you look at this picture here, and you see that the volatiles HCNN are now landing in the magma ocean, completely molten mantle with a magma ocean. A lot of it goes into the core. A lot of these elements partition into metal. They go into the core. So probably 90% of Earth's water and carbon reservoirs are kept in the core today. So you've got to understand this partition between magma ocean and core before you can understand at all what comes out into the atmosphere. That's sort of a geochemical question, so I'm very interested in geochemistry now. What happens then is when this planet stops accreting, then the magma ocean crystallizes. So the magma ocean crystallizes, it becomes solid from the bottom up, as it does so. It spews out all of these volatiles into an atmosphere. And then the question is, what does this atmosphere contain of? It can either be reducing, or it can be oxidizing. It can be H2 and CO rich, or it can be H2O and CO2 rich. Mars likely outgassed an atmosphere that was rich in H2. That's great, but H2 escapes with the solar irradiation, and so Mars doesn't have an atmosphere today, probably because it outgassed a lot of H2. Earth outgassed an atmosphere of CO2 instead, and so did Venus. That's why we could maintain our atmosphere today. Because we don't have 100 atmospheres of CO2 today, luckily. A lot of it most of it got rained down through the oceans and mixed into the mantle again. So in that case, the ordinary life would have to wait until CO2 precipitated into the mantle. So much CO2 would surely be poisonous to the cells. So one possibility is actually that Mars might have better conditions for the origin of life early on, because Mars has an H2-rich atmosphere. Lightning in an H2-rich atmosphere can make biomolecules. And so it's a possibility that Mars had the better conditions for the origin of life, but then Mars lost its atmosphere. Maybe the life would then somehow transfer to Earth rather than continue to survive. Or maybe there's a way for the origin of life to have occurred on Earth directly. One thing we are very excited about in the field right now is this little thing here, the JWST Space Telescope. Very expensive and very successful. It is now doing its first observations. And one of the goals of JWST is to measure rocky exoplanet atmospheres, so the atmospheres of planets around other stars. So one of the things that I'm very excited about in the next few years is to get to know about the first CO2, H2O, CO measurements of planets around other stars. And we want to know about this because we want to understand what are the conditions for the origin of life in other planetary systems, and this will eventually help us answer the question of how common could life be in our galaxy. So, thank you.
Very exciting. Yes.
So if Martian Earth formed at the same time from same type of pebbles, why do one have H2 atmosphere, which can be gas out, and the other one has a...
It's actually a geochemical question, because it has to do with the amount of oxygen in this magma ocean. So Mars is only 10% of the mass of Earth. Mars is much smaller and had a lower pressure. So Mars's magma ocean has far less O2 in it. So Mars is outgassing this H2. On the other hand, Earth has an effect in its magma ocean where it gets very oxidized, a lot of oxygen in it. And that oxygen also gets outgassed and combines with H2 to make H2O. So, and the same with CO and CO2. So it probably has to do with the mass of the planet. And again, nothing is random in my view. Mars was doomed to lose its atmosphere because it was doomed to outgas H2. And Earth had to maintain its atmosphere because of the deeper magma ocean. It would
so in principle,
CO2.
you think you can calculate it within some years at least?
Yeah.
Well, it's created a lot of questions. Unfortunately, we have time for 2 more questions. One there and then Per.
I wonder about oxygen and do you think that oxygen appeared before origin of life or after as a consequence of photosynthesis?
No, it's definitely a consequence of the origin of life, oxygen, but there's a different level. There's an oxidation where you have free oxygen. Then there's another chemical oxidation where it's about whether you have H2 or H2O or CO or CO2, not free oxygen, but just the amount of oxygen. And that's why we say the Earth's early atmosphere was oxidizing. I mean, it had a lot of CO2. It doesn't mean it had O2. In order to have O2, you need free O2. You need a biological process simply to release a lot of it, right?
The next question is then, can you look into these exoplanets and see you can detect free oxygen?
Yes, you can definitely.
Do you do that?
You can look for CO2, you can look for oxygen, you can look for CO. And problem is that free oxygen may be the consequence of destroying H2O by UV radiation. So if you destroy H2O by UV at the top of the atmosphere, you make a fake oxygen that's not a biomarker. So it's probably going to be scrutiny of interpretation, what it means.
Okay, Per, I'm sorry, that would be the last question.
So you said that the water came with comets to Earth, but then why are the comets icy and where? So how did they become?
Yeah. Okay.
Water?
Okay. So, I mean, in the old model, it comes sort of with the comet, and we think it comes with the small pebbles. But the idea is that as you go away from the sun, it gets colder and colder, and close to the sun or water, there's the same amount of water, but it's all water vapor. It cannot become accreted beyond 1 AU or something. Water becomes ice, and it goes really 30% ice. So comets are 30% water. Earth is only 1,000 ppm or when you count over the whole object, right? So it has to do with, there's water everywhere. It has to do, if you want to treat it, it has to be solid. It cannot be water vapor. It's way too volatile to capture.
Thank you very much again. And now Beren Martin, sorry if I didn't pronounce it. She's professor in organic chemistry at Stockholm Universitet, and she got the prize in chemistry in 18, sorry.
Thank you very much.
Thank you.
Yeah, so thank you. I want to start by thanking the Göran Gustafsson Foundation for first giving me the opportunity to continue my research, and also for giving me the opportunity today to be here and tell you a little bit about what we are doing in my laboratory. So today I will talk about catalysis for sustainable organic synthesis. And I want to start by first thanking all the people that have done most of the job that I will be very briefly commenting today. So I'm very thankful to my group, very motivated students. I think for me, the best thing about being a professor is to have the opportunity to work with young and motivated people all the time. I think we have a privilege in this situation. Of course, I also want to thank all the funding agencies that have supported our research, as well as key collaborators within the academia and also within the industry. So what I do is to create organic molecules. Organic molecules are composed by carbon, hydrogen, and many other heteroatoms, oxygen, nitrogen, fluor. And all of these heteroatoms, together with the molecule per se, give these specific molecules a specific property. For example, we have here many molecules with different biological activity. Many of them are pharmaceuticals or used for the construction of other materials. But how can we make these molecules, these complicated molecules, effectively? So many of the research that I will discuss today focus on introducing these heteroatoms. We consider oxygens, nitrogens, and iodine, bromide, fluor containing functional groups, and these are very important because these specific functional groups give the specific properties, for example, if we are thinking about pharmaceuticals. But what tools do we have to put all these atoms together in the correct place and to be able to use them for different applications? So an important factor that we have to think about when we are trying to make these molecules is also the selectivity. So you have some sort of starting materials. We have got many starting materials from fossil resources, but we need to elaborate them up until we get to these complex molecules. So how can we do that selectively so that we don't waste our resources? And also, lately, what we are focusing a lot on is on how can we replace those fossil resources by other sustainable resources to see if we can get to this very same type of compounds. So selectivity in the processes that we make is very important. So we do this, we introduce these functionalities by using different catalysts. And I have used many catalysts based on transition metal complexes and also organocatalysts. Those are at the left side of the screen. I have also used a lot of heterogeneous catalysts, and these are based on materials, for example, on metal organic frameworks or silica materials or metal foams that can also mediate specific reactions. But this is not something you come up with like that. You need to start somewhere, and you need to start studying the mechanism of the whole transformation to identify each intermediate during the reaction process so that you can then improve the process or develop new processes. So we combine this with a lot of theoretical chemistry investigations, experimental investigations in the laboratory, and we try to follow the reactions in situ so that we can see what's happening as the reaction proceeds to see if we can improve the catalyst efficiency. So catalysis is everywhere. Absolutely everything we produce today, I can say most of it requires a synthetic step where a catalytic reaction is needed. For the synthesis of pharmaceuticals, as I mentioned, constructing these carbon heteroatom bonds specifically in a specific position can only be done with the help of a catalyst. Also, agrochemicals, for example, for the synthesis of ammonia, which is used to produce fertilizers, and that's why we can feed the whole population nowadays. But also to make polymers, there are of course catalysts in the catalytic converters in our cars, in the food industry. So catalysis is everywhere. So we need to develop catalysis, but we also need to develop it efficiently. As I mentioned also before, so we are working in the red square where we have materials, we convert them to highly functionalized materials that are useful for the industry and for consumers. And then we work a lot on trying to recycle all the resources that we have used, and also on replacing fossil resources by other sustainable resources. So, very simple how a catalyst works. So we have a compound or starting material that we need to transform into something else. So we have to overcome a certain energy. So we have to go through a transition state. So if we can use a catalyst, the minimum advantage we are going to have is that we will require less energy. So we can go again from the semi-starting material to the final product using less energy. But also, there are some specific processes that without the catalyst, this wouldn't happen at all. So catalysis is important to create these highly functionalized molecules. And I will just give 3 examples, 3 short stories, I can say, of how we have been using catalytic processes and how we are trying to make these catalytic processes more sustainable. So the first type of example is dealing with the late stage functionalization of very complex organic molecules. So let's say you have a drug, such as that one that you can see in the slide, and this drug is used for the treatment of cystic fibrosis. So let's say you want to develop a new drug. Let's say you just want to vary a small part of this molecule by putting a functional group, let's say a heteroatom or something. So making this new drug from scratch will take a lot of synthetic steps that need to be optimized, and that it will also prolong the time to get to that specific molecule. Then you get to the molecule and then you need to test it, and then you may have good properties or bad properties. So there is a lot of effort and resources to get to a derivative that you don't even know, or you can predict to some extent, but you don't know if it's going to be useful or not. So what we've been trying to do in the last few years is to develop catalytic processes that can functionalize these various complex molecules. So this is what we call late stage functionalization. So it has to be a chemoselective reaction. So you want to attack a specific point in that molecule, not any other point on the molecule. And you want to create a sufficient amount to be able to do some tests. So this is extremely important in drug discovery. So within this area, we decided to work on the CH functionalization. So this is now you can look at the other molecule on the right and you can see all the carbon-hydrogen bonds. There is a lot of carbon-hydrogen bonds in each molecule. So what we wanted to do was to functionalize one of the CH bonds selectively. So it gives a lot of opportunities because there are so many CH bonds in the molecule, but of course it's also a challenge to just specifically attack one of these CH bonds. So usually what we do, we work with some model compounds to develop the catalytic process. And we started working with these benzoic acids. The initial idea behind this is also because we were developing methods to transform products derived from lignin degradation. So to be able to use bioresources towards the synthesis of aromatic compounds. So we came up with this catalyst, which is an iridium complex. And this iridium complex was able to break this CH bond that I highlighted there in blue, and with the proper ingredients, transform it into a carbon-iodine bond. So this worked very well in the model reaction. However, we had some problems on selectivity. So if you had 2 identical CH bonds, you could not just functionalize only one of them, but you got functionalization of both of them. So we went forward, and by just studying the mechanism and changing the conditions, we developed a method that now gives us only one functionalization. Even if we have two identical CH bonds, now we are able to tackle only one functionalization. So this is selectivity. We have not lost the material. We can choose if we want to put one atom or two atoms just by modifying the catalytic system. The reaction is extremely selective, as I show in that graph there. So then we turned into changing a CH bond for a C-methyl bond, introducing a CH3 group. This is because the so-called magic methyl effect, which is a phenomenon that has been observed in medicinal chemistry, that the addition of a methyl group can have a tremendous effect on the pharmacological properties of the molecule. So if we are able to take a molecule and introduce simple and effectively a methyl group, we have created a derivative that can be used for further testing. So we use a source for the methyl group was this boron potassium tetrafluoroborate. We use the same type of catalyst, which is a very simple iridium catalyst, and we can introduce these methyl groups that are highlighted in red. But now the question is, okay, we do all this chemistry in model compounds, but is it really useful for complex systems? So we then turned into the late stage methylation of different drugs by using the iridium system that I presented in the previous slide. And the answer was yes, and we were very happy to see this. So if you see the methyl group, which is highlighted in green there in the screen, is introduced always next to a carboxylic acid. So it's always introduced next to the same functionality. So we only attack a specific CH group, and this is good because you create a single molecule in a single step with insufficient amounts to be tested for biological activity. If then this derivative is useful for medicinal chemistry, then perhaps a new synthetic group needs to be developed. But we give access very quick to different drug analogs. So then we also studied the properties of these drug analogs, and then we could see that the methylated compounds compared to the non-methylated compounds had an increased metabolic stability, as we observe a decrease in the intrinsic clearance. So we also then tested went on to further develop this chemistry, and we introduced a carbon-nitrogen bond, and this gives us an amino group, and amino groups are also present in many compounds with biological activity. So we could do that using also the iridium system, and then we also tested this in the late stage functionalization of many drugs. Later on, we also expanded the concept, so we could introduce these functionalities not just next to the same functional group, which is in blue, the carboxylic acid, but then we could also vary different functionalities. The second example, this is the first approach towards a sustainable catalysis, as we can make a very complex compound in a single step from something that is already available. The second example I want to give is presented here, and this is also an iridium complex. It's a little bit more sophisticated, so we had to do a lot of mechanistic investigations to get to that structure on the top, this bifunctional catalyst. And the reaction that we were studying is shown there on the top right-hand side. So we have an amine, the nitrogen compound, that we then alkylate using an alcohol. So this, in comparison with known traditional methods, this is a very sustainable atom economical method because it produces water and the only byproduct. So the reaction per se is very clean, but then you need to have a catalyst that works very well. So our first generation catalyst enabled us to do this reaction in the absence of any other additive. So we didn't need base or anything else. So that is already an increase in sustainability. However, our studies revealed that in the mechanism you had an iridium intermediate, which is there in the brackets, and this is a very stable compound. What happens here is that, okay, then we need to develop the second generation of the catalyst that would avoid forming such a stable species. If in the mechanism you form a very stable species, then you need much more energy to go to the next intermediate. So we developed the second generation, which is that one with a nitrogen ligand on it, and now we could do exactly the same chemistry at room temperature with no base, quantitative yields. So this is by doing studies, mechanistic studies, you can develop the structure of your catalyst so that the reactions are quantitative and clean. And this, of course, is very important for sustainable development. We use this catalyst for the functionalization of amino acids. So amino acids, if you want, they are important building blocks, for example, for the synthesis of peptidomimetics. If you want to transform them, they are difficult substrates to work with. First of all, what you have to do normally is to protect this OH group that is there. Then you do a transformation in the nitrogen, and then you deprotect. So it's not sustainable at all because you have to go through 3 different steps just to get one compound. So with this catalyst, what we could do was to do the transformation without the necessity of introducing and deprotecting and introducing and removing different protecting groups. So we have quantitative yields, and these are just some of the examples of the compounds that we can make. So the reaction is quantitative, the product precipitates, and it's very easy purification. So we are, again, attacking sustainability by reducing the number of synthetic steps as well as facilitating the purification. One problem we have here, we use an iridium complex, and iridium is very scarce. And so if we use it, we have to use it well. We need to recover and we need to reuse it. So the good thing with this catalyst is that it also can be recycled many times. So if you create a new catalyst, it has to be active enough, but at the same time stable enough so that you can recover it from the mixture and reuse it, ideally infinite times. So this is the first step towards that. And this brings me to the very last example of sustainable catalysis. And here what we studied was the incorporation of hydrogen into molecules. So there we have a triple bond that it will form a double bond. So typically this is done with palladium as the catalyst and using hydrogen as the source of the hydrogen atoms. So hydrogen comes from fossil resources, it's also highly flammable. We also need organic solvents, and the palladium catalyst is a noble metal, scarce, and also pyrophoric. But this functional group that we create is, of course, also very important for many. We can find it in many complex organic molecules with important biological activity. So what we did in this case was to try to get rid of palladium, try to get rid of hydrogen. So now instead of hydrogen, we use water. So we produce the hydrogen directly from water. And instead of using palladium, we used a nickel foam that you have in the picture there in the middle, which is an abundant metal. So with this, we could also reduce many triple bonds. We could also use deuterated water to form deuterated compounds, also of importance in medicinal chemistry. So now, where are we going now? We are very concerned about sustainability, and we develop reactions that we believe they are more sustainable, but are they really more sustainable? So now we go one step forward, and what we try to do is to do a prospective life cycle assessment of the whole process. And of course, the industry is very used to do that for large processes, but we don't apply this into the academia. So one wonders also, why would you develop a catalytic reaction if this is not going to be really useful because it's very expensive, it's not sustainable, etc. So what we are trying to do is to transfer this knowledge now to the academia so that whatever we develop new in the laboratory has can be followed up all the way, and so we can develop sustainable catalytic processes. So with this, I just want to thank you for listening and for the opportunity for being here. Thank you.
Thank you so much. We have time for one question. Anyone have a question? Yes.
I was sort of wondering, biology must be great at having discovered catalysis for 4.5 billion years. How often do you find something and realize this is better than what the cells are doing? Or do you sometimes see this?
Not very often,
No?
I would say.
No.
Of course, nature is our best example to follow. Also, all catalytic processes in nature are highly complex, so you don't have a single catalyst working towards making one reaction. You have 20 catalytic cycles working together towards producing something. So this is extremely difficult to mimic in a laboratory.
Well, thank you very much. We go to our last speaker, one of our prize winners here, Hans Ringström, professor in mathematics at KTH, Royal Institute of Technology in Stockholm. You got the prize in mathematics in 2011. Welcome.
Okay, so to finish some light entertainment, mathematics. Okay, so let me start by thanking, first of all, the Göran Gustafsson Foundation for funding my research, but also to the organizers for inviting me. I mean, it's a great pleasure to be giving a talk at this anniversary celebration. So I want to talk about some results I obtained while being funded by the Gustafsson Foundation. And the basic question here is, what is the shape of the universe, right? So this is the question, right? And now, to be able to address this question properly, I'm going to have to go through some background material. And now I will, in fact, spend most of my time on background material, so general relativity and topology. So let me start with one slide about general relativity, right? So general relativity is a geometric theory of gravity. And the path to geometry starts already with special relativity. So in fact, special relativity is most concisely summarized simply by one geometric structure. So this is Minkowski space. I've written it here just R4, eta. R4, these are just 4 tuples of real numbers, and eta, well, this is some geometric structure. Let's not go into the details of that, right? Okay, but now we have a problem because special relativity is not consistent with Newtonian gravity. So in Newtonian gravity, the speed of propagation of the gravitational field is infinite. This doesn't go very well with special relativity, so we have to have a new theory of gravity. And in his search for a new theory, Einstein was guided by some principles, particularly the so-called equivalence principle, and some very ingenious thought experiments. And this led him to the conclusion that gravitation should really correspond to some sort of distortion of this geometry, this Minkowski space type geometry, right? And so the conclusion here of general relativity is that space-time is really a geometric object. It's a so-called Lorentz manifold. So it's a manifold M with a Lorentz metric G. And then you ask the question, what is a manifold and what is a Lorentz metric, right? Okay, so here are some very simple pictures, right? So these are two-dimensional manifolds. These are examples of two-dimensional manifolds. And what is the characterizing property of a manifold? If you take a point on a manifold, there must be a local, in the neighborhood of that point, there's some sort of coordinate system. You can set up an x and a y axis to sort of measure the coordinates. And you should be able to cover the entire manifold with coordinate systems like this. And then we sort of make a transformation from the perspective of one coordinate system to another. Then this transformation should behave in a nice way. This is the sort of defining property. And these are all examples of this. Now, due to this construction, you also get something called a tangent space at each point. So if you take a sphere, right? If you take the north pole of the sphere, then there's a natural tangent plane there. So it's just a horizontal plane, right? And the point is, in a manifold, you always get these tangent planes, right? So now what is a Lorentz metric? Well, if you take a 4-dimensional manifold, you have these tangent planes, and they're just like R4, like we had in Minkowski space, right? It's R4. And then you can put this geometric structure like you had in special relativity. You just put them in each tangent space. And then they should fit together nicely, right? So this is then what a Lorentz manifold is. Now, the last 3 examples here, well, the one at the bottom, they're a little bit special. They're going to play an important role in what follows. They're said to be closed manifolds. So what does this mean? They don't have a boundary. You don't reach the end. And no matter how you view it, you can't sort of escape to infinity, right? So this is the notion of a closed manifold. So now you know what Lorentz manifolds are and what closed manifolds are, and we can go ahead, right? So now let me sort of have a one slide explanation of the distinction between topology and geometry, right? So from a topologist perspective, a sphere and an ellipsoid are the same thing. But from a geometrist perspective, they're not the same thing, right? So the point is that in topology, you can take an object and you can sort of continuously deform it. You do not change the topology, but you change the geometry, certainly, right? And so the point is that, I mean, on a 2-manifold, which is like topologically a sphere, you can put infinitely many different geometries, right? So the point is, there is a chance of classifying topologies, but you can't classify geometries. That doesn't make sense, right? Okay, so now we can return to our question, right? What is the shape of the universe? Well, the universe is, as I said, it's a Lorentz manifold, so it's a manifold which has a Lorentz metric. So the shape of the universe, it's really this M, this manifold, right? And now you can ask the question then, it has to admit the Lorentz metric. And does that mean that there are restrictions? Well, yes, there are restrictions. So if you look at these examples now, the first top ones, they admit a Lorentz metric, this geometric structure that we have to have. The sphere does not admit such a structure. There is no Lorentz metric on the sphere. That's excluded. If you look at the torus here, it looks like a donut. This does admit a Lorentz metric, this type of structure, but for every Lorentz metric you put on this thing, you can travel to your past. This is not something we want to have, perhaps, right? So we want to exclude those types of shapes. And the last one, again, there is no Lorentz metric on this object, right? Okay, so the conclusion here is that the fact that space-time must admit this geometric structure, a Lorentz metric, this entails, I mean, a significant restriction on the possible shapes of the universe, right? And in particular, if this M is closed, well, then every Lorentz metric on a closed manifold is such that you can travel into your past. You want to exclude that, right? And in fact, if you make rather natural, but I mean slightly technical conditions I don't want to go into here, the manifold, the shape of the space-time must be sigma cross R. So what do I mean by this? So you should just think of it, it's just like the cylinder, right? So in the cylinder you have circles as cross-sections. Here we don't have circles as cross-sections, we have some general three-dimensional manifold as cross-sections instead, right? And, okay, this is slightly subjective, but from my point of view, it's natural, in cosmology at least, to think that sigma is a closed manifold. So again, no boundary, you can't escape to infinity, right? Okay, and I should also say that this division here, you can really think of this as a topological division of space-time into space and time. There's no canonical geometric division of space-time into space and time, but there is a canonical topological division into space and time. So you can think of R as sort of parametrizing time, and you can think of sigma as sort of parametrizing space, right? So now you end up with a question, okay, so what can we say about closed three-dimensional manifolds? So that all the shape now in this object is really in this closed three manifolds. So what can we say about this closed three manifold? The remarkable thing, amazing to my mind, is that at the beginning of the present century, closed three manifolds were classified. This is, to my mind at least, one of the most impressive results in all of mathematics. So, and to explain this classification in all detail, this is a little bit technical, but I want to give you a flavor of this amazing result, right? So, okay, so let's take a three-dimensional manifold. So sigma is now a three-dimensional manifold. Now, if you have a three-dimensional manifold, then you can write it like this. Okay, it doesn't. The resolution is not fantastic. Okay, but okay, so you can write it like this. And now you ask, okay, what do all these symbols mean, right? So let me start then by discussing what does the hashtag mean? What does the symbol hashtag mean? Well, hashtag denotes connected sum. So the operation is the following. So if you take two manifolds of the same dimension, what you can do is you can cut out balls in each of them, and then you can sort of identify the corresponding spheres. And this produces a new manifold, and this is called the connected sum of the original ones, right? So this is, I've tried to illustrate this operation in the bottom. If you take sort of two tori, do this thing, and then you end up with this object on the right, right? Okay, so now we know what hashtag is. And you can think of this operation, hashtag, you can think of it a little bit like a product, right? So if you take, for instance, if you take a positive integer, right, then you can factor it uniquely into a product of primes, right, up to order. And for closed three manifolds, it works a little bit the same way, right? So this connected sum operation, you can, well, so you can think of this, what I've written there at the top, like a prime factorization of a three manifold. You take a closed three manifold, it has a unique decomposition as a product of primes. So these are then sort of, these things between the hashtags, these are elementary constituents. So prime manifolds, right? Okay, and finally then you ask the question, what is this S3 divided by gamma 1? Let's not get into that somehow. These are like elementary manifolds which have been known for ages, so let's not get into that. And then S2 cross S1, this is sort of also sort of elementary stuff. But the KIs, this is a little bit interesting. These are also well understood. So there, for the KIs, you have to, you actually have to do something called a torus decomposition. This is more complicated. Let's not get into that, right? Okay, so now, right. So now it's interesting to compare this with standard cosmology, right? So in standard cosmology, you assume the cosmological principle. So this is sort of the idea that at one point in time, you can't distinguish between 2 points in space, and you can't distinguish between 2 directions. And if you make this assumption, what restrictions do you get on this division? The restrictions you get is that this decomposition has to be trivial, first of all. There can only be one of these factors. It could be an S3 divided by gamma I factor, that's okay. It cannot be one of these S2 cross S1s, that's not allowed. It could be one of these KIs, but if it is a KI, this torus decomposition, it also has to be trivial, and of the 6 constituents possible, only 2 are allowed. So the message I'm trying to give you here is that if you assume, sort of make the standard assumption like you do in cosmology, the cosmological principle, then you've excluded all the possible shapes of the universe before you even started comparing with observations, right? So this is sort of what okay, this ends the introduction, and now we can go over to the actual talk, so to speak, right? So now let's return to this question. What can we say about the shape of the universe, right? Okay, we have to make some sort of assumptions to get conclusions. So let's make some assumptions here. So first of all, let's assume that the observational data indicate that to our past, the universe is well approximated by one of the currently preferred models, the ones preferred by cosmologists today, right? That's a reasonable assumption to make. Let's assume that if we interpret the data in this model, we see back a certain time. We don't see all the way back to the Big Bang, but we see, I mean, for instance, if it's electromagnetic radiation, I mean, we would see back to the time where you have decoupling between radiation and matter or something like that. So you can think of T0 as representing, for instance, decoupling or something, right? Okay, and then there is a Big Bang. Again, this is what the cosmologists are telling us. And then the last assumption, this is a philosophical assumption, right, which you often make in cosmology. You make a very strong assumption to be able to draw some sort of conclusion. And this is the statement that analogous statements apply to all observers in the universe, right? With, I mean, the same, that they see the same model, right? Okay, so, and this, I mean, this is a philosophical assumption. This is not something you can test observationally, but this is the kind of assumptions you often make in cosmology, right? Okay, so these are, to my mind, very strong assumptions, and I don't really see that you can make stronger assumptions, that it would be reasonable to make stronger assumptions. And then the question is, what conclusions are we now allowed to draw concerning the shape of the universe? And the answer is no conclusions, right? Right? Okay, so the point here is that if you give me a closed three-dimensional manifold, sigma, right? You give me a standard solution, and this is how the universe is supposed to appear, right? If you give me also a distance to this solution, you fix some epsilon, and also a norm with respect to which I'm going to measure the distance. And you give me also this T0 that I mentioned on the previous transparency. If you give me all these things, what will I give you in return? What I will give you in return is a solution to the Einstein equations with appropriate matter content and the shape you prescribe to me, sigma cross r, right? The solution will have a Big Bang, and for each observer in the space-time, what they see to the future of T0 will be epsilon close to the standard model you prescribe to me, right? So in this solution that I construct here, you cannot, I mean, effectively, you cannot tell the difference between the solution that I construct and the standard solution that you prescribe to me, right? So sort of to summarize the whole thing, so if you use the cosmological principle as a starting point, this is going to exclude essentially all the possible shapes of the universe before you've even made any comparison with observations, right? If you assume that all observers in the universe consider the universe to be close to one of the standard models, then you have no restrictions at all, right? And so you can ask the question, do you really want to insist on this principle? Okay, so So again, thank you to the Gustafsson Foundation for funding this research, and thank you for listening.
It's almost a bit like Hitchhiker's Guide to the Galaxy, right? What was the answer to 42?
42, yeah.
Exactly. But maybe someone has a better question. Yes?
Sometimes we hear from string theories that there may be other theories of gravity, and that GR generatively may not be more than approximation. How does that change the mathematical view of?
Well, I mean, so I'm a mathematician. I'm a mere mathematician, so to speak. So I'm just working in the framework of the general theory of relativity. I mean, of course, if you have other theories, then things might change, so to speak. I mean, I should say it's very strongly dependent on the fact that the current preference is really for flat, right? I mean, so you currently prefer flat models, right? I mean, if the preference would be, for instance, for that the universe is close to spherical, so k+1. Then you would get conclusions. So then there would be restrictions. But yeah.
So yeah, I mean, there is this classification universe k less than 1, bigger than 1, and equals 1. So you say if it's bigger than 1, that's when you have a closed universe, right? Yeah, am I right?
Well, I mean, okay. So the point is that the current
have a closed universe, because then you can go back.
I mean, so the currently preferred, well, thanks Ari Gubler here, so to speak, right? I mean, but currently people prefer, there should be some dark energy, right? Yes. And usually it's a positive cosmological constant, so to speak, right? And this positive cosmological constant causes this very strong expansion of the universe, right? And interestingly enough, this actually makes a mathematician's life much easier, because that's a much less subtle problem to deal with, actually, when you have all this expansion, right? So even you can have solutions with k equals 0, +1 or -1 that still expand, all of them, right? If you put lambda equals to 0, then what you would get, k equals -1 is going to expand, k equals 0, then it's going to expand, but marginally, and k equals +1, you're going to get rid of that. So that's sort of the classical picture before, I mean, people became convinced that, universe started to expand in an exponential way recently, so to speak.
Yes.
Hans, I was once on your promotion committee.
Yeah. Okay. I have to watch out there. Yeah.
And I studied your material quite a lot. I would not say that I understood all of it. One thing that struck us in the committee was that you had not graduated very many PhD students, and we asked about that, and you said that the problem with the PhD student is that they don't understand what I'm talking about. But we were still quite impressed by your work and how you described your work. And you were promoted to professor. You also later got the Göran Gustafsson Prize. But I'd like to ask you how you have used this. I understand what you're talking about is kind of complicated, even if you try to make it extremely simple. But how have you used this money if it's so difficult to get students to understand? Because most of us use the money to pay salaries for students. Did you use it just to avoid administration, to leave you alone?
Well, I certainly did use it for my own salary, right? I mean, that I have to admit, right? I mean, and that's what contributed to solving this. But I mean, of course, I also have PhD students that do solve interesting problems and so and postdocs.
Avoid administration by keep them away from you. This is one of the major problems that the Swedish universities, that the professors have to do on this. What you call it.
Well, I think it was a great series of talks and presentation, very broad variety in science, which we are so proud of being able to support here in the Gustafsson Foundation. Now we'll have thank you. 5 minutes bensträckare. Okej, let's go to Swedish. Nu tar vi en 5 minuters bensträckare Men vi kommer samla upp några mer ståbord här och paneldeltagarna är välkomna att komma fram i. Medan ni andra antingen bara står upp eller sitter kvar eller går ut vad ni vill. Men om 5 minuter kör vi igång igen. Jag börjar med att presentera våra panelister här. Jag är väldigt glad att ni har kommit hit. Det är då Kristina Moberg, ordförande i professor i organisk kemi på då Reses på KVA.
Tidigare.
15 till 18.
Ja.
Och även pristagare. Och sen har vi Göran Sandberg, professor i skoglig genetik.
Det räcker.
Rektor på Umeå universitet 2005 till 2010 och sedan dess verkställande ledamot på KVA. Knut och Alice Wallenberg stiftelsen. Samuel Engblom, jurist och statssekreterare i utbildningsdepartementet med ansvar för högre utbildning, forskning och frågor. Tack för att komma. Jag vet att det är väldigt bråda tider för alla statstjänstemän just nu. Och sen har vi Hans Ellegren, då, professor i evolutionsbiologi och ställningssekreterare på KVA sedan det här året. Alltså jag hade förstått att man är ställningssekreterare, men egentligen är man bara en viss tid. Så vi ska prata lite om liksom, betydelsen för privat forskningsstöd och även då betydelsen av sådana här forskningspriser. Och då tänkte jag börja med en pristagare, Kristina, som fick det då 1998 i kemi. Liksom, speciellt dina tankar över just betydelsen av sådana här vetenskapliga priser. Och kanske också privat enskilda priser gentemot att man får ett team för diskussion.
Naturligtvis är det en väldigt stor ära att få ett sånt här pris. Att få ett pris överhuvudtaget och också ett erkännande av det som man gör. Gustafssonpriset är ju speciellt också, liksom en del andra priser, med att det också är en stor prissumma för forskning. Vilket har haft oerhört stor betydelse. Därför att det ger en möjlighet att göra den typen av forskning som är väldigt svår att göra annars. Man har pengar som är i beredskap. Man behöver inte söka. Jag kommer ju från KTH och där har man inte fakultetsmedel för studenter. Utan man är helt beroende av extern forskning. Hittar man då till exempel en postdoktor som är väldigt meriterad som man vill knyta till gruppen. Så får man söka pengar och det kanske tar ett halvår, åtminstone flera månader innan man får dem där. Med de här prispengarna för Gustafsson så var det alltså möjligt att rekrytera direkt. Så det hade en väldigt stor betydelse. Men sen så tycker jag att det finns en annan aspekt på vetenskapliga priser. Att de också betyder, åtminstone de stora vetenskapliga priserna. Nobelpriser framför allt, men även i viss utsträckning andra priser. Att de annonseras ut. Det står i dagstidningar och så vidare. Och att fira vetenskapen. Att forskare kan framstå som några som har skrivit om i tidningen. Det är inte bara artister, idrottsmän och så vidare. Och det tycker jag är väldigt viktigt för vetenskapen också. Och det är en aspekt tycker jag som man inte ska glömma bort när man pratar om vikten av stora vetenskapliga priser.
Ja, tack. Bra tankar. Och det ska jag göra när man Knut med Wallenberg stiftelsen är ju faktiskt en av Europas allra största privata forskningsfinansiärer. Som har över 100 år på nacken. Så jag misstänker att du har försökt fundera mycket på vilken roll den privata finansieringen har gentemot det offentliga. Och liksom, finns det risk att staten kanske minskar sitt ansvar om ni gör för mycket?
Alltså staten står ju för 85% av finansiering av forskning i Sverige. Jag tror vi står för 6-7%. Så staten är ju förarsätet och måste vara det. Men jag hävdar att vi gör saker bättre inom vissa nischer än vad staten kan göra. Vi är oerhört mycket mer snabba till beslut när det är kris. Vi var först i covid. Det tog 2 dagar för oss att avsätta 250 miljoner. Sen kom de statliga myndigheterna. Sen är vi strikt elitistiska på ett sätt som man inte kan vara i en offentlig finansiär. Och det är klart, vi kommer att dela ut 30 miljarder de 10 år som kommer. Så vi kommer att vara de största finansiärerna av fri forskning i Sverige. På ett sätt som jag inte ser hos de statliga finansiärerna nu. Jag är orolig över att de statliga finansiärerna blir mer och mer fragmenterade. De blir mer och mer kortsiktiga. Och de kopplar mer till innovation än till grundforskning och utbildning. Så jag tycker att vi har en väldigt tydlig nisch.
Tack, ja. Och jag ska fråga Samuel då på hur du ser på det här och liksom just därför det privata versus det statliga. Ja, liksom ni olika staten och privata olika roller och mål. Och jag säger liksom, hur ser departementet liksom på en långsiktiga forskningsfinansiering i Sverige?
Ja, först ska jag bara tacka för att jag fått komma hit. Jag var på högtidssammankomsten här i våras, så då var då jag fick upp ögonen för Göran Gustafssons stiftelsen och just den här konstruktionen på priserna vi pratade om här tidigare, där det ger väldigt mycket pengar. Och det är ett bra exempel på en viktig privat forskningsfinansiär. Sen har vi ju Wallenbergstiftelserna i Sverige som givetvis gör, som du säger, som inte är ju väldigt stora om vi tittar på europeisk botten och är jätteviktiga. Jag tror så här, när det gäller samspelet som redan nämns är den offentliga finansieringen i särklass störst, och staten måste liksom alltid göra sitt. Vi har ett ansvar att se till att alla typer av forskning som behöver finansieras kan få finansiering i någon form. Det innebär då att så här, var hamnar vi då? Det innebär att det finns mycket forskning och utveckling som bedrivs i industrin, och den är ju ännu större än den statliga liksom, forskningsfinansieringen. Ja, det som typiskt sett ska hamna där, ja, det är inte det som vi då ska finansiera med våra statliga forskningsmedel, utan det är ju annat, och det ska vara grundforskning. Sen så vet jag just det, så diskussionen drar staten iväg från det, och det är liksom att vi har nu tillsatt en utredning för att se över forskningsfinansieringen, den statliga forskningsfinansieringen, både själva hur den är organiserad. Det handlar ju om att vi har ett antal olika finansiärer och hur många ska vi ha i framtiden? Ska vi ha fler eller färre, och vad ska de ha för inriktning, och hur det ser deras arbetsformer ut också. Hur ser merfinansiering är en sån sak som ligger i den utredningen. Sen så tror jag när det gäller liksom hur den privata finansieringen och offentliga kompletterar varandra, så tänker jag att ibland så är det ju så att vi kan se att det är bra. Jag tror mångfald i forskningsfinansiering är alltid bra. Och att det då finns andra finansiärer som gör andra prioriteringar staten gör. Det är väldigt viktigt. Därför att då är det forskning som kan få finansiering som inte får i det statliga systemet av olika skäl. Det andra är att ibland så lyckas vi gå samman och satsa liksom gemensamt, göra gemensamma viktiga satsningar på strategiska områden. Och där jag tror att staten utmanas av det, alltså då, när till exempel Wallenbergstiftelsen kommer och säger vi är beredda att satsa på det här, och då kan staten hamna i ett läge där staten är beredd att möta upp. Så där finns en kombination. Sen så tror jag att också i de tider vi lever i nu, en viktig roll för privat forskningsfinansiering, det är att finansiera sånt som en stat kanske inte vill finansiera av politiska skäl. Vi har sett situationer där det har varit maktskifte någonstans, och man som en politisk skäl så drar man in viss finansiering. Och då har vi sett exempel på privata finansiärer där har gått in och sagt vi täcker upp den här. För eftersom den här forskningen då anses politiskt kontroversiell. Och det har vi sett i USA till exempel. Vi har i Europa exemplet med, jag tänker, om jag går tillbaka till mitt gamla område då, jag är jurist, disputerad jurist i botten. Om vi tittar på i Östeuropa, så till exempel Open Society Foundation, George Soros Foundation, de har finansierat forskning som regeringar i de länder de verkligen inte alls har varit förtjust i. Och det har varit väldigt viktigt för den forskningen inom statskunskap, juridik, mänskliga rättigheter i de länderna. Så det tror jag också är en viktig sak att komma ihåg när det gäller den privata finansieringen.
Ja, tack för de synpunkterna. En annan viktig fråga är liksom universitetets roll i det hela. Hur universitetet liksom ska styra och komma in med forskning. Och Hans, du har ju liksom skrivit artiklar om det här. Universiteten är inga vanliga myndigheter, har du skrivit. Och så hur ser du på universitetets roll som myndigheten inte var inne och det hänger ihop med privat finansiell forskning i relation till det då?
Ja, jag tycker det finns två stora trender när det gäller forskningsfinansiering i Sverige. Det ena är att den politiska styrningen av inriktningen på forskningen ökar för varje forskningsproposition som kommer. Jag var in på Vetenskapsrådets hemsida häromdagen, och bara på en av de olika ämnesråden fanns det 17 olika utlysningar med mer eller mindre smala områden. Och det är ändå vår fria forskningsfinansiering. En annan trend är ju att universiteten själva rycks med i det här. Man är så angelägen, och man också krävs att vara goda myndigheter. Just. Man har rektor som myndighetschef. Man har väldigt kompetent och ambitiös personal på förvaltningen som lite grann ges utrymme att styra mycket av verksamheten. Och man vill helt enkelt vara duktig på sin administration. Och allt det här kommer ner på att det blir mindre tid över för forskarna att driva forskning. Det blir smalare liksom möjligheter. Om det då finns finansiärer som Göran Gustafssons stiftelsen, så får de bästa forskarna en sorts fri möjlighet. Och vi har hört exempel på att direkt kunna få en idé. Om man behöver rekrytera en ung person som plötsligt dyker upp, då måste man ha de här fria pengarna. Och det är just där som de privata forskningsfinansiärerna är så viktiga för svensk konkurrenskraft och för, kan man säga, återväxten inom svensk forskning och svensk näringsliv.
Vilket jag tror också, fri forskning är så viktig. Och kanske staten håller med. Skulle inte staten kunna gå in och ge lite mer pengar till fri forskning då? Man sätter upp statlig fond som man låter organisera ungefär som Göran Gustafsson eller Knut Wallenbergs stiftelse.
Alltså, om man ska svara på det.
Ja, du får svara någon annan först.
Du kan börja svara. Vi styr ju inte universiteten på det viset som man styr andra myndigheter. Utan så den här, det som är effekten du får är ju att på grund av medfinansieringsregler till exempel, så är det så att mycket av det som ska vara de fria medlen används istället för att möta upp extern finansiering. Och då gör det så att de prioriteringar som görs för externa finansiering får ju ett genomslag tillbaka på lärosätena och hur de använder. Det är ju liksom en förstärkande effekt. Och det har vi då i den här utredningen faktiskt tittat på det här med medfinansieringen. För den får liksom, jag är medveten om det, och det binder upp ganska mycket av de pengar som man har. Så det är ett sätt att liksom, det är en sak som vi kan se över. Sen ska vi komma ihåg att jag menar, mycket av det som går till exempel vetenskapsrådet är ju tänkt att det ska vara sånt som är så att säga, som reaktivt, som jag läste. Jag tycker det var ett dåligt ord, men det var kanske en bra beskrivning. Det vill säga att det är pengar som finns att söka utan att det är sagt till vad. Så att det är klart att det finns ju fortfarande mycket pengar till exempel vetenskapsrådet för sånt som ju är friare och som också är på sån här så att säga, excellens ska gå till excellent forskning. Så att jag vill inte gå med på beskrivningen att liksom allt som går till VR är uppbundet. Men det stämmer ju att man liksom, vi försöker ju rikta också forskningsmedel mot vissa samhällsutmaningar. Och ibland är det en förutsättning för att vi ska få de pengarna. Vill säga.
Ja, alltså de som framförallt kräver medfinansiering, det är ju de statliga forskningsfinansiärerna. Alltså vi har ju blivit anklagade med i år så betalar vi snitt 23% overhead. Och snittoverheaden för forskningsuniversiteten är 29%. Sen kan ju inte vi då för om KTH drar iväg och ska ha 45 eller KI, det är en annan sak. Det är ju myndigheter som ligger under er, statliga myndigheter som kräver medfinansiering av statliga universitet. Och jag tycker problemet, det finns tillräckligt med pengar i svensk forskning. Men det är för spritt på olika finansiärer som var och en uppfinner sitt eget begrepp av vetenskap och kvalitet. Jag kan ju inte fatta att VR och Formas och Forte och Vinnova har olika definitioner på vad vetenskaplig kvalitet är. Det är det första. Det andra, det är att vi har ett universitetssystem med för många forskare och för dåligt ledarskap. Nu råkar vi ha en rektor här som dessutom har varit en utmärkt forskare. Och det är ju ett föredöme. Jag hävdar ju bestämt att ska man leda ett stort universitet måste man kunna kärnverksamheten. Antingen om man behöver inte vara en världsklassforskare, men man måste förstå processen, eller också om man har varit en bra pedagog. Nu ska jag inte recensera alla rektorer i landet, men det är klart att kan säga att det är inte alla som faller i någon av de två kategorierna. Så jag tror inte att du behöver fixa mer pengar. Jag tror att ni bara behöver fixa mer struktur och stringens i systemet.
Men jag tänker, det här visar liksom på dubbelheten. För några av de sakerna du tar upp, det är ju sånt som lärosätena faktiskt styr själva. Absolut. Man väljer sina rektorer, sen så stämplar regeringen det, kan man vill säga. Också det här bestämmer hur mycket overhead man ska ta ut. Det är ju beslut på lärosätets nivå. Men jag håller med dig om, och det har vi lagt in i den här utredningen också, det här med medfinansieringen. Därför att det får de här effekterna. Sen är det också någonting som faktiskt också stoppat in där. Det handlar om forskarnas administrativa börda. Det här att kunna, som du säger, att har man då väldigt olika definitioner på saker eller man ställer väldigt olika krav, det är olika saker som ska fyllas i på olika sätt. Då drar vi upp forskarnas administrativa börda. Jag tycker ett bra exempel nu att de här som då inte riktigt fick EU-pengar, att de får en chans att få andra pengar till exempel utan att ska göra nya ansökningar. Lite sådana lösningar för att lägga mindre tid på att söka pengar och mer tid på att forska. Det tycker vi är väldigt bra.
Men det har ju skett en väldigt stor förändring under den tiden. Det var ganska lång tid som jag har varit aktiv inom forskningen. Går jag tillbaka till min början, när jag inte bara var en väldigt ung forskare, så hade jag betydligt mera medel. Jag kunde använda fakultetsmedel att avleda doktorander. Jag hade hjälp, jag hade sekreterarhjälp. Det var väldigt mycket jag inte behövde göra. Allt sånt här försvann sedan. Det fanns inte möjlighet till fakultetsmedel. Jag tror att en orsak är naturligtvis den ökade administrationen och ökade overheaden. Men också väldigt många krav som universiteten måste sköta. Och där kommer väl dina argument att det inte bör vara myndighet. Och sedan lokalhyrorna som är naturligtvis ett väldigt stort problem också. Väldigt mycket pengar går. När man pratar med utländska forskare så tror ju de att det är guld och gröna skogar i Sverige. När man tittar på mycket som avsätts till forskningen. Men det är inte riktigt så. Därför att det är väldigt dyrt att bedriva forskning här. Våra doktorander kostar mycket. Bra i och för sig att de har bra löner. Men det går åt väldigt mycket stora anslag. Får man miljonanslag så låter det väldigt mycket. Men det är en doktorand man har då.
Ja, får jag bara fylla i det. Så för vi som arbetar på golvet, Kristina och jag som har varit forskare och vet hur det ser ut i verkligheten. Visst är det så att det går lite för mycket tid att skriva ansökningar. Och det beror i grunden på att det finns för många personer i systemet. Men det är så mycket som har förändrats under de senaste decennierna. Du har varit med lite mer än mig än jag har varit. Men det har ändå skett en stor förändring. Alltså det är så mycket som är myndighetsrelaterat. Eller det är kultur som har vuxit fram av sig själv inom universiteten för att man vill vara duktiga. Vare sig man är en myndighet eller inte. Då går det så mycket tid i vardagen att sitta i det ena eller andra kommittén eller göra den ena eller andra sammanställningen. Rapportera, allt det mätbara ska mätas. Och så ska man så säga hur många handlingsplaner man har för det ena eller andra. Just att den här fragmenterade vardagen för forskarna. Att hon eller han inte får tid till att tänka eller att prata forskning med sina kollegor. Det är det som gör det svårt för den svenska konkurrenskraften.
Ska vi ha flera? Ja.
Alltså, nu ska jag inte försvara politiken, men man kan konstatera, det var ju en annan regering då, men i alliansen ökar ju forskningsbudget extremt mycket. Vi har något som är huvudmannarådet med rektorerna och chefen på akademierna. De bestämmer ju våra anslagsformer och så. Och då ställde en av mina chefer frågan, vad gjorde ni till rektorerna när ni fick så mycket mer pengar? Och 4 av rektorerna svarade, vi anställde mer folk. Det verkar som man kunde inte tänka tanken att man använder pengarna till att låta de bästa forskarna få bättre resurser som inte behövde göra där. Universiteten har ju liksom byggts ut fenomenalt mycket. Och jag fattar precis att plocka bort de 15% som aldrig levererar på ett universitet, det går bara att göra genom någon typ av svältdödsstrategi. Det går ju aldrig att få bort dem eljest. Men jag tycker också att man måste, nu tittar jag på en rektor här, du har inte varit så länge. Men jag tycker också att universiteten och kollegiet måste ta ett visst ansvar till att det är på det här viset.
Men så kanske jag också säga att om det finns några frågor från åhörarna här så får jag slänga upp en hand. Ja, där var Arne.
Jag tänkte på den här utredningen som du pratade om. Uppenbarligen den här balansen man ser mellan basanslag och grundforskningsanslag via VR eller andra hur mycket som ska gå till andra myndigheter, den andelen. Hur kommer man, har ni några idéer på vilken riktning det skulle kunna gå? Eller är det...
Så här, den utredningen ska se över alltså den statliga externa forskningsfinansieringen. Så den ser ju inte över liksom, basanslagen. Sen hur mycket man lägger var, det är ju liksom en budget. Liksom, du kan ju ha ett system och sen kan du välja hur mycket pengar häller du i olika delar av systemet. Så utredningen kommer ju titta på systemet givetvis. Men vi tar ju upp, i direktiven finns sådana här frågor som till exempel medfinansieringen. Och det är klart att då har man en anledning att titta på vad det får för effekt för basanslaget. Men sen blir det ju en fråga, liksom, var kommer vi kunna lägga forskningsresurser? Och det blir ju en fråga inför nästa forskningsproposition egentligen, hur vi liksom kalibrerar de här olika typerna av finansiering. Så.
Jag förstår att ofta så är det ju svårighet för regeringar att lägga pengar till mer obundna satsningar som på projektanslag på vetenskapsrådet, som många forskare älskar förstås, eller kunna söka den. Den delen har ju vuxit ganska långsamt över åren, det senaste decenniet i alla fall. Medan då, infrastrukturmedel har ju vuxit ganska mycket och det har ju varit väldigt bra i och för sig. Men från regeringens sida, politiska sidan, är det väl lättare att titta på strategiska satsningar, alltså strategiska områden politiskt lättare att sälja, förstår jag.
Ja, alltså, ska du sälja in? Jag tycker ändå det är ganska intressant med forskningsmedel, för att det är någonting som människor är beredda att satsa på. Vi är beredda att satsa på det politiskt. Vi har också mycket av de här, det finns ju många liksom, privata donationer som kommer från människor som kanske haft ganska lite kontakt med forskningen. Jag pratade med styrelseordförande i Lund där tidigare i år som berättade om att de hade fått donationer från människor som hade haft kontakt med universitetet. Men de var övertygade om att forskning var någonting i grunden bra och det ville de bidra till. Och jag tänker så här, vi måste, det är klart att det är lättare för en extern publik att sälja in, eller ett finansdepartement eller vad det kan vara, att sälja in liksom forskning där man kan säga att men vi behöver forska om det här för det ger den här avkastningen liksom på ganska kort sikt. Det svåra är ju hela tiden att säga att sen är det så här att det finns massa forskning vi måste bedriva som vi inte vet vad den kommer leda till. Men vi måste bedriva så mycket sån forskning att när någonting händer, som till exempel en pandemi, så finns redskapen där. Och pandemin är ju faktiskt ett bra... Det finns ett case man kan göra utifrån det. Att det var en del saker som gick till... Och det finns även en del kring den här nya gröna tekniken som är sånt som har gått från grundforskning till tillämpningsforskning väldigt fort. Och vi visste inte vad det var tidigare. Och det är ju det som är det pedagogiska liksom, tricket. Hela tiden kunna förklara. Och så vill vi att ni ger massa pengar till sånt som ni inte förstår och som ni inte vet vad det kommer gå för nytta till. Men om vi tittar historiskt så har det här varit bra för samhället att göra så.
Absolut, jag hade en synpunkt eller fråga. Jag tycker att forskningsfinansiärerna ska ställa mycket högre krav på effektiviteten när deras pengar används. Ni pratar om att universiteten vill gärna vara duktiga och det är inte frågan om det. Det är frågan om att rädslan sprider sig. De är så rädda för att göra fel så att det kostar otroligt mycket pengar. Och jag tycker att forskningsfinansiärerna borde lite grann ta bladet ur munnen och säga det. Det finns ju ett antal olika områden där rädslan är större än annat. Själv håller jag ju på mycket med försöksdjur och det här är ju nästan helt absurt. Alltså, ni pratar om att det tar tid att skriva ansökningar. Det tar ännu längre tid att skriva djurförsöksetisk ansökning. Det är en fruktansvärd tid. Och det har ingenting med etik att göra. Det är ren forskningsfientlighet. Och jag tror att man måste highlighta det. För snart har vi ingen sån här typ av experimentell forskning i det här landet kvar. Det är så dyrt att göra så att vi kan ju inte... Det spelar ingen roll om vi får mycket pengar från er om vi inte kan använda på ett effektivt sätt. Jag tror att man måste lyfta den diskussionen. Det här skulle vi spara pengar på.
Någon som har någon kommentar på det?
Om jag pratar om våran sida, jag förstår inte vad vi ska göra åt det. Jag ser ju många i det här rummet som har fått oerhört mycket pengar av oss. Och jag tror inte att det är någon här som tycker att vi har jagat ihjäl dem med rapporter och pekpinnar. Vi kan ju... Vi ger pengarna, sen hoppas vi på det bästa. Vi kan inte göra annat. Sen finns det, vi kan inte lägga oss i alla regelverk som finns. Alltså min förra chef, Peter Wallenberg, han sa en sak som jag tog till mig väldigt hårt för 13 år sedan. Vi utför inte forskningen. Forskningen utförs av forskare på universiteten. Vi ska vara extremt noga med att komma med pekpinnar. Och då kommer vi inte med pekpinnar vad man ska göra. Men vi kan heller inte komma med pekpinnar till rektorer och andra hur de ska bete sig. Sen är det en annan sak inom den statliga sektorn. Den vet jag för lite hur den fungerar. Men i vårt fall har vi varit supertydliga på det.
Ja, det som herr Holmberg säger, alltså det är i grunden är ju politiska beslut som har lett fram till exempel det här med försöksdjursregelverket. På samma sätt som det gäller med etikprövning, överklagandenämnden, där det finns de som idag är rädda eller vågar inte belysa vissa frågeställningar. För de är så rädda för att det är kontroversiellt. Många samhällsvetare nämner det här. Och det är i grunden politiska beslut som försvårar för forskarna att bedriva sin verksamhet. Återigen, hur mycket pengar man än stoppar in.
Jag tänker det sista du säger här just om, alltså, jag har blivit uppmärksammat att det har vi gjort. Alltså det handlar ju om skärpningar i regelverket men också en ny struktur vad gäller det forskningsetiska. Och där har ju, och då har det hänt ett antal saker, ett antal uppmärksammade fall som ju då utifrån det som man kan liksom läsa om dem framstår som är ganska underligt. Och där har vi sagt, dels så gav vi ett uppdrag där till ansvariga myndigheter att liksom förbättra sin information. Och sen Anna Ekström sa det i ett svar på en debattartikel i DN häromdagen just om att vi faktiskt är beredda, då är vi beredda att se över de här reglerna om det visar sig att det sätter liksom, hinder för, så att det kräver helt enkelt sånt som inte bör belastas med den typen av prövningar, eller att vi får de här väldigt konstiga effekterna där man har. Och det är ju det här med en sak i forskning, alltså jag förstår kritiken att man har tagit ett regelverk som handlar om forskning, egentligen medicinsk forskning framförallt, och så tillämpar det på många andra områden. Men så det är vi där är vi beredda att se över. Det är ett nytt regelverk och det är en ny liksom, myndighetsstruktur kring det.
Jag tror att en allmän tendens är också att man har inte tillräckligt stort förtroende för forskarna. Det tycker jag är väldigt viktigt. Att man tror att forskarna vet vad de ska göra. Jag har varit i styrelsen på forskningsrådet i Danmark i 8 år. Och där, speciellt i det forskningsrådet och grundforskningsfonder i Danmark, så har man ett väldigt stort förtroende för forskarna. Man säger ofta, istället för att man har en massa föreskrifter om hur saker och ting ska göras, så säger man forskarna vet bäst hur det här ska göras, för de förstår det här. Det är de som ska göra det hela. Och det är väl lite grann det du säger om Wallenbergstiftelsen också. Medan däremot till och med inom en del stora satsningar inom vetenskapsrådet, så måste man rapportera med. Det finns vissa kringkriterier som måste följas. Och det förtroendet tycker jag att man saknar ibland.
Ja, och det tycker jag det är lite samma sak. Så jag tänkte, om staten använder en stor pott med pengar, så används det till fri forskning och man ger ut pengar ungefär som Wallenberg ger ut pengar. Man kan söka om vad man vill och så. Ja, då riskerar att inte kan styra forskningen, men då kanske det blir en absolut skarpast. Oberoende av vilket område det är som får pengar att forska lite.
Alltså vetenskapsrådet har ju den rollen. Det ska ju vara den typen av forskning. Sen är det här, ibland så är det här identifierar vi en viss samhällsutmaning. Där behöver vi göra, liksom rikta forskning. Och det är liksom det som exemplet du nämnde, att er förmåga att snabbt rikta om pengar till specifika problem, den förmågan måste ju även staten ha. Så det är ju inte fel att vilja styra forskningsmedel. Men det är klart, vi kan inte styra alla forskningsmedel. Det är det som är viktigt. Det måste finnas en tillräckligt stor kaka som forskar på det här som vi inte förstår och som vi inte vet vad det ska användas till. Men något av det kommer att vara så värdefullt i framtiden att det var värt alla pengar och lite till.
Jag tror att det du föreslår, det vore en dröm, men det är en utopi med en stor grundforskningsfond. Jag tror alltså att det är 5 eller 6 forskningsberedningar. Och det här har man ju diskuterat länge. Men det finns ju ett politiskt spel, alltså ni måste ha argument för det. Om jag fick önska någonting på era planerade sidor, att var inte så klåfingriga med vetenskapsrådet. Alltså de behöver inte ha 2 sidor med utlysningstexter. Man kan låta dem släppa dem lite mer. Men det svenska systemet måste jobba ihop. Vi är ett så pass litet land och vi är ganska bra på det. Vi kan ta Welcome Trust i England och regeringen i England. De jobbar jättenära varandra. Medan, jag ska inte nämna något specifikt land, men det finns ju ett land i Norden som har väldigt starka privata fonder. Där det finns egentligen ingen direkt dialog med rektorer och så vidare. Och jag tror då blir det en fara om den privata forskningsfinansieringen blir så stor att man börjar styra forskningspolitiken. Utan den privata finansieringen måste gå hand i hand med staten och med universiteten. Men samtidigt så måste man erkänna att vi måste ställa kvalitetskrav. Vi ställer för lite kvalitetskrav både på universiteten och i det svenska finansieringssystemet.
Tid ute, några sekunder. Det tänkte vi alla avsluta med att säga liksom, vad de önskar skulle ske en enda forskningsmässigt. Göran, det är ju svarat, tror jag. Ja. Så det går från Hans och sen Samuel och sen Kristina. Kort, vad skulle du verkligen önska?
Jag replikerar Kristina. Jag tror att politiker behöver ha mer förtroende och tillit till forskarna.
Det är extremt byråkratiskt. Jag hoppas att den här utredningen vi har tillsatt ger oss ett underlag för nästa forskningspropp och för att komma till rätta med några av de sakerna som man nämnt.
Okej. Kristina, sista ordet. Som första.
Att förstå vad den grundläggande forskningen är viktig. Och det tror jag att politikerna är lite rädda för det. De vill gärna att det ska vara så stor nytta som möjligt med pengarna. Och det är inte alltid som största nyttan kommer när man bestämmer precis vad som ska göras. Den oväntade nyttan har vi som tema här på KVA.
Tack ska ni ha. Den har alltid väldigt fort. Jag skulle också säga tack till KVA som liksom varit värd åt oss här. Vi får låna Beijersalen. Och tack för den hjälp som ni ger stiftelsen hela tiden för att utvärdera de kandidatvinnarna ni ska ha i styrelsen. Och ja, tack för oss. Slut för idag. Nej, det var inte alls slut. Break för idag. Om 2 timmar hoppas jag vi flesta av oss ses nere på Stadshuset. Där är då välkomstring från Fru och 48 då intas borden.